Hello, hello.
Hi, good morning.
Good morning. Good morning, distinguished delegates, respected speakers, and participants. It gives me immense pleasure to welcome you to the third day of our international workshop. Over the past 2 days, we have engaged in critical dialogues on marine litter mitigation, biodiversity conservation, and climate change challenges, supported by valuable regional and global perspectives. Today, we culminate this knowledge exchange with a dedicated masterclass segment, a hands-on, technically intensive series of sessions designed to equip participants with advanced tools and methodologies pivotal to contemporary marine spatial and biodiversity research. So to commence today's proceeding, we have the first segment of our master class focusing on the spatial mapping of marine debris using remote sensing technologies. This session will be conducted by Dr. S.K. Dar, Scientist F at NCCR, who will introduce advanced methodologies for detecting, tracking, and analyzing marine debris distribution patterns through remote sensing application. Dr. Sisir Kumar Das, Scientist F, whose expertise in optical oceanography and remote sensing had advanced our understanding of ocean carbonic fluxes and coastal dynamics. His work in satellite-based monitoring of sediment transport, erosion, and fishery support vital management efforts. Now, over to you, sir.
Good morning. So, can we start? Okay. So, welcome back to the third day of this international workshop on marine litter, climate change, and biodiversity as a global challenge. So, I think last 2 days we are discussing about different type of problems, issues, observations, different aspect of marine litter, climate change, and biodiversity loss. Uh, for everybody, just I want to tell one thing that when we plan this program, this masterclass theory, actually people should do actually prior to conference one pre-training but this is a uniqueness that it has been done after that. We are thinking to show you some demonstration that what type of liters through remote sensing technology you can do, what type of spectral indices it is available. So the outline of my presentation, I will give a short presentation. Along with me, my student Ms. Gayatri is there, Dr. Nimalan is there, then Sujit is there, and Dr. Maya is there. So I think we will share this 1 hour into different parts. So can you open the slide? When we are talking about marine litter, the first things comes to that what we visualize is a macro litter. But over the time, very recently, people are doing macro to micro to nano. So many intensive research has been done over the last, I will say, 5 to 7 years. So remote sensing is one tool. I think we need to understand very closely that how spatial mapping of marine debris from remote sensing can be attempted. It cannot give you the exact picture, but you need to attempt because every time you cannot go to field, collect the samples, analyze it. It will take enormous effort. So whereas remote sensing can give you at least a holistic picture about that where is the debris is available. Next, 1 minute. Okay. So marine litter is always giving an always affecting the ocean health. It is through food chain and the pollutions what is coming into the ocean that lead to again economic impact. So all these disasters is, I will say it is a small disaster type of things is happening when pollution comes to your environment. So the best effort is how to prevent this. You need lot of awareness programs, you need to monitor periodically, you have to know when a tsunami comes or a flooding is happening, it carries lot of debris from the river to the ocean. So this type of debris when it is there, when it interact with tide, wave and everything, so mostly everything will come and deposit in the near to the coast. So that can cause lot of damage to the ecosystem. So for that purpose, I think with the modern technology, using different type of advanced technology like remote sensing, machine learning, deep learning, we need to see that how best we can quantify this type of marine litter in the environment. If you see the conventional method, what you do remote sensing, mostly it will tell you direct measurements, what you can see. Like if you see you can see the chlorophyll patch, there is a algal bloom is happening. From the ship you can see that, and that also you can monitor through satellite as well. So whatever direct visualization can be possible, like ship is moving, ship track, ship trail, all these things you can do visual monitoring. And apart from that, what is not visible, you can take some approximations and you can convert into different type of parameters, like sea surface temperature, your dissolved organic carbon, dissolved inorganic carbons, all these things you can do by certain hypotheses or certain algorithms. So that mostly remote sensing in an oceanic environment, when you analyze, you do that type of work. The limitation is always that cloud is always there for optical, and it has the resolution makes an important role, because you cannot point a target, but however it will give you a picture of, it will give you the information of a particular area or particular site. Good thing that you need not to spend lot, you can analyze a satellite image, so labor, cost of labor and equipment will be reduced, all these things. And even also you can do the change detections, that means how over the period of time it is changing, all these things can be assessed, can be easily assessed. Now the question is, that's why to overcome all these problems, remote sensing is the best approach, where accessibility is there, holistic view is there, your minimal disturbances is there, hotspot identifications, everything can be possible in a single picture. So this is not new, because if you see from the science ocean color 77, when the CZCS was launched by NASA. So during that time, that with a high spectral resolution of almost 1 kilometer, so there are a lot of observation, Earth observation satellites were there, and it can track lot of bloomings, lot of particles and everything. But since then, since 1977 to 2025, the technology has been changed a lot. That means it has improved significantly the spatial resolution, where you can go now centimeter level data sets also available. It is India also having similar type of sensors. So you need, even there are a lot of type of sensors like microwave sensors is also looking into this. So if you go that, if you see overlook into that, there are two type of sensor, as you all know that as a basic of remote sensing. I think all of you are everybody is a biologist, or anybody is remote sensing? Boys, please range who is working on remote sensing. 1, 2, 3, 4, 5. Very good. So this class will be relevant to all of you, I think. Okay, so very good that that remote sensing is a very nice tool, where you can take the data sets from different sensors. Don't ask. I know you must have a question in your brain. Anytime if you want to stop me, stop. Raise your hand and please stop me. I will answer. Then after that we will go ahead. Okay. So because this is a master class, we need to interact. We need to hear. It is not like the end of my lecture you will ask questions, then I will answer it. You can interrupt when if you are not understanding anything. So don't worry about, when I am talking about remote sensing, you don't worry about accuracy, what will be the accuracy, how it will estimate, how marine litter can be quantified. Somebody might be thinking, I think Dinaga is thinking, sir, you are telling when the marine litter comes, it will come as a numbers, right? How many objects are there? Whereas remote sensing can give you the exact number of marine litter available in a particular area. Yes, that answer I will give you in the end of my presentation. But yes, if you carefully go through different type of data sets from different sensors, then at least you will get a holistic idea that exactly where actually hotspots are available. That means where maximum litter accumulation is happening. So first is to identify where we need to target. You need a very high resolution data. When I am talking about high resolution, what will be the resolution? You might be thinking. Okay. So when high resolution, in the sense, it will be in meter to centimeter level. That means if somebody is going on a field, and if you are physically measurement with a particularly 5 meter by 5 meter square on the beach, or in ocean, if you are taking one particular lagoon system or a smaller area of, let's assume that 1 kilometer by 1 kilometer area. So particularly that area, how many samples you will go and collect. So you will collect at least by maximum 10, 20 or 100 like that. But whereas in one picture of a satellite image, you can see that really litter is available or not. And if you take the series of satellite image, if you target that every day or every alternate day, that means every 2 days once, at least you can see that how the movement is also happening. So this type of things is there. So for that, you need not to be that in cloud free seasons. If it is cloud is there, you can avoid passive sensors, but if cloud is there, you can take the active sensors, because active sensors will do what? It will penetrate through cloud and it will give you the realistic pictures. So there are sensors like radar, synthetic aperture radars are there, lidar is there, sonars are there. So all these sensors are mostly you will find in the active sensors. In passive sensor, mostly commonly used is radiometer, and that we have seen many missions, as I said, Landsat, that is 1977 onwards, 1 to till Landsat 8. I think Sentinel, Prism, Worldview, 2x3, all these things are available. Indian remote sensing satellites also, lot of sensors are available. You might be thinking, sir, this is okay, where you will get the data? I will show you, don't worry. So all these questions, whatever when I am talking, please write it down so that in the meantime you can ask me also. So the main objective of remote sensing is that it can detect, it can do 4 things. One, detection, it will tell you the categorization, it can categorize what type of litter it is. It can, overall it can tell you how much area or how much quantification also you can do if it's possible. Then also in a periodic, if you repeated images, if you can analyze, you can tell that how it is tracking, how this movement is happening. So all detection, categorization, quantification and tracking, all these 4 things you can do, which from a synoptic observations or your in situ data you cannot do. You can only, you can do one task at only one time. Okay, modelings. Yesterday we were listening Eric, and he was talking about lot of ghost net modelings and everything. So you can try, so that is that projections how do. So this input you can take also, you can verify whether really it is moving or not. So a lot of validation exercises can be done with model by using from the satellite data, and satellite data also can be validated by using the in situ data. So it is so one is in situ data, what you analyze in the lab, one is remote sensing and one is modeling. So these 3 things are interconnected each other. I think we need to understand all these things very critically. So the first is that detection. When I am looking into detection, how the satellite image look like. Everybody might be thinking that, you might have seen, I think everybody nowadays using Google Earth images in your mobile and everything, right? So you have seen a area, if you zoom it, you can get some pictures. So some pictures I am just showing you. This is all my first presentation will be based on the review of literatures, what worldwide people are using, people have got analyzing the satellite images. And last part of my presentation I will show that what NCCR and what we are doing our own research. So from satellite, if you see, or any Google Earth image, if you see, it mostly used for detection purpose. So remember, all pixels when you analyze, you need to analyze very carefully. It is all pixels, if it is written black, white, green, anything is there, all pixels are not carrying the litter information. It is, you need to understand that which pixel is a litter, that need to be verified from the ground truth. Then after that you can analyze, you can confirm that yes, satellite can also measure and detect the marine litter. So certain things are I am showing here, and some pictures also has been taken from drone. because remote sensing has a limitations of that though we talk about data, mostly 1 meter resolution data is freely available. But rather than otherwise centimeter level data is not readily available. So people are using different type of drones so that they can get very high resolution data sets, and from that they can analyze different type of machine learning algorithms and optical algorithms. All these things they analyze and they get the required input. So if you see what type of sensors are available, the most nowadays popular is that Sentinel-2, Worldview-2, Prisma, that is a hyperspectral. Hyperspectral sensor is nothing but a sensor which is having a enormous bands, some minimum 200 bands will be there or more than that. So all these bands, when it is there, what does that mean? What do you mean by a band? Spectral band. Spectral band is nothing but, you know, the electromagnetic spectrum, where it is starting visible from 400, it starts with 700 nanometer, but after that it will go NIR band, IR band, all these things will be there. So each band you will get one information. In hyperspectral you will get, I think, sometimes 500 bands also you will get. And so people, many researcher, they use these hyperspectral bands for detection purpose and as well as your monitoring, regular monitoring and every tracking purpose also they use this type of data sets. Landsat data sets are also available. So I will show you that where you will get. I think I request all of you, please take this photograph, this snapshot of this, otherwise end of this presentation you can get it. Now this is the sources where you can get the data set freely. So you can download this, you can go to the each link and then you can download your requisite informations where the satellite data is available. Okay? I think everybody is taken? Okay. Now, satellite data is available, I have given you source, but as I am talking about the drone, are the drone data sets is available? No. Drone data sets are very limited, but still Google Earth image platform, I think Dr. Maya he will explain that how you can get this type of high resolution pictures. But nowadays drone, if anybody want to get any images, how you will analyze, how you will classify, because you need to classify, you need to identify that what type of litter it is. And precisely speaking, you cannot, through a remote sensing, you cannot characterize each and every litter, but you can characterize maximum how much plastics can be there, how much styrofoams will be there, that type of things can be assessed. But you cannot get the total numbers of a single frame, because drone image it is quite possible, because we use people are using many machine learning techniques, artificial intelligence, so from that you can get it. But mostly drone images are again I will tell you it is having a limited applications. Now regarding this, this is some drone sensors. When you are putting drone, it is not like a simple giving RGB image. There are different type of sensors also you can put it in your drone, and what is his objective, that also need to be defined. Suppose lidar based, if need, because when you track you need a GPS system also as well, should be embedded into your drone, because it will tell you that exact locations and what type of litter, everything. So this type of sensors you can, this is also you can take a snapshot for your reference. So each instrument, that means I am talking about sensors, they have their own resolutions, they have so that is mostly available, and we also use some sort of similar type of drones for our studies. Now, now let us come to a little bit basic of remote sensing. So basic of remote sensing, what is happening? It is only it is between as I told you, electromagnetic spectrum starting from 400 to almost you can go for IR, which is 11.3 to 12.3 or 12.5 to 13.5. So beyond that, up to 2500 nanometer also you can go. So if you see that in a traditional way, what they do actually, if you see classifications, you aggregate, if you take all litter into a machine, it will pass through a light, correct, and it will tell you that what type of litter. Somebody is connected. Oi? So microphone is on, I think. Okay. Yeah. So you pass through a one light, and when you light, it will take the spectral signature of each object. So what does it mean that spectral signature is nothing but the maximum reflectance what you are getting from a target. So different, as we know that there are all objects in the world, each and every object behaves spectrally different. Their spectral signature is different. Not a unit, not two identical. Suppose two type of color, black, if you put it in a single source of light, you will find different spectral characteristics. So sometimes you need to see that what type of litter actually I am targeting. Suppose you are quantifying only the plastics. The plastics is again different type of plastics are there. Hard plastic, soft plastic, PET bottles, all these things. So if you take all the plastics category and put in a single source of light, and if you want to compute the spectral curve, it will be all will be different. Not only that, even the chemical composition of plastic, what is available, it is not that. Even it will determine different type of colors are painted on that. Suppose this, let us assume this is a glass. This glass, if I will put it as a blue color, and if I will measurement I will do, I will get a single one curve I will get. Now if I will wash it, and again if I will put another red color on that, on the same glass, my spectral characteristics will be different. Correct? So now question is that ultimately we know that there is a single glass which is a liter. When it is broken, we will throw it and it will become a liter. But how you will analyze this, how you will quantify this, that is a big challenge. So for that purpose you have to jump, artificial intelligence. You have to train your models, you have to put lot of data sets into that. It depends upon remote sensing. Actually, I think many basic remote sensing there will be many type of classifications are available. It can tell you shape, size, texture, okay, so your symmetry, all these things are matters a lot. So you need to analyze if that, suppose this glass, if I will put it, yesterday I was talking to somebody, some group of people, if I will take one glass bottle, okay, glass bottle with full of water, if I will put it and I will take a picture on my mobile, I will get a picture, right? Now same size of glass bottle, if I will take one PET bottle and I will put the water, and if I will take a picture, can you differentiate which one is glass and which one is PET bottle? It is quite difficult. But the human intelligence comes into picture there again. PET bottle, when you put the water, okay, it definitely, if you see minutely, if you observe, there must be some shape difference will be there. Correct? Or the texture of the bottle is different, it's not so smooth bottles will be there. So when you can detect, then a sensor can detect. Because human intelligence only, always I feel that if that whatever your eyes can detect, then you can, then definitely one photograph also can detect the same amount of information. Yeah.
So, is there a method to try and detect transparent objects? Because do they even have any reflective?
Yeah, again one more thing, transparent object. When you are taking a picture, you are depending on a light source of your own sensor, that means own mobile, or a external light, correct, which is sun or something like that. So when a light you are putting on a glass, you, most of the light will transmission will happen. Whereas the styloforms of a similar space, styloform is there, the light will not pass through that. So that spectral signature again will be different. So it is quite difficult to do things. So that's why that's why we are coming into the artificial intelligence, where you need each and every type of liter with all combinations. It is not like that only one single line wood is there, the spectral signature, if I will apply into my satellite image, all will be wood. No. You need permutation combination. Suppose you are targeting 10 liter on a particular area, that I am going and quantify 10 type of liters. So what I have to do, 1 to 10, 10 factorial if you take, it has, you need lot of combination will come. And all these combinations you need to put it on your classification methodology. Then whatever the output will come, that can be a realistic figure. But now next question is, sir, how I can do, being a student or being a researcher, how I can do all these permutation and combinations? Because you will not get a liter whenever, if you go see a dump yard or in a beach, you will not find a accumulated one type of liters only available. You will get mixed of all these things. So critically analyzing all the spectral signatures is very, very important. So, and when we start our research, we thought we will, instead of going quantification directly, let us analyze this type of spectral signature, get ready for that, then after that we will analyze. I think I will show you all our activities. 30 minutes over. Then after that, there are many literatures, if you see, people use different type of band combinations. Why band combination is required? See, I told you that each spectral wavelength will give you one type of spectral signature, correct? So if you take multiple bands, two bands if I will take, one band, let NIR band or IR band. This NIR band again, it is not, I have not mentioned about what is the lambda. So if I will take, let assume 708 nanometer or something like that. So particularly one wavelength and another IR wavelength, if I will take that, if you make a ratio, what we will get? We will get a normalized differentiated vegetation index. That is mostly we do for in the land, but in the water you cannot classify like this. So in water, when you do, we assume that these liters are a physical things which we can see directly. So we need again the help of NIR band and IR band. But if it is submerged in the water, if it is mixed with the water, this band ratio will not work. Whatever you can visualize, whatever a picture can visualize, that pictures only you can, if you make any band ratio, you can do it. Suppose such turbidity I am talking, Turbidity is what? It is nothing but the total suspended solid concentration. Suspended solid concentration, can you see that? No. Now if I will add if I will make this water is very clear, if I will make turbid, can you see that? You can see the turbid water, but can you see the particle size? No, because it is already mixed. You can see when you take into a microscope, correct? Yeah, but your normal eye cannot detect that. That means 400 to 700 nanometer you cannot get these things. So for that purpose, what you need different type of approach, spectral analysis like NDVI, plastic indices, floating debris indices. There you can quantify easily that this is the total patch of floating debris, but it will not tell you that what type of debris it is. It will tell you that what is the nutshell, what is the floating debris is available on the image. So this type of equations are available through various researcher. Nowadays modifying versions is also coming, because people have taken lot of data sets and they modified all these different type of bands, because when you analyze, when you do that, I think that this particular tables will tell you all those references. The different type of algorithms are available to classify your image spectrally. Similarly, you can, beyond that, you can do machine learning techniques, different type of classification, random forest classification, SBM classifications, all are available. So that also you can try. But before trying, again I will tell you that all the inputs, whatever you are providing to the image, you need to understand the science behind that. Without understanding that type of signatures, spectral signatures, you cannot, because the in machine learning also it will take the combination of all this permutation combination it will take, and it will tell you that. So I am very grateful that today this Andriolo is coming online. Yeah. So he has actually done extensive work. I think he is joining up today. I think you will interact, you can ask this type of questions to him. You see, in a beach, a particular place, how different type of litters can be classified and can be done using drone images. He has taken some drone images, and it is classified different type of litters also. So finally the output will be like this. Your final output will be like this. So easy. So but the question is that drone images getting every locations, positions, type of things, observations, everything depend on you where you will get this images. This data set is not freely available. You have to fly a drone with a particular sensors. So capability of drones is possible, to get, even we also fly some drones, and we are also trying to get different type of litters. There is another work which NCCR has initiated with the association with Jamstack, Japan, where the capabilities of drone on marine litter classification can be done. It was machine learning, artificial neural network techniques. It was a little quite impressive, because if you see one picture of a beach, still picture, using different type of models, machine learning techniques, you can classify and you can characterize what type of litter it is, whether it is a glass, it is a plastic, or it is a wood. So this type of techniques are there, different type of softwares also there. So people are walking into that, but don't again ask me what is the accuracy, okay? because accuracy again depend on the analysis methodology. So that is, we are trying to improve, the scientific community is trying to improve the algorithms and everything, but it is possible. Why I want to tell you that it is possible to characterize, to classify still pictures, okay, from your camera or drone or satellite, anything, classification can be done. Quantification, this is one smaller area, but if you take a bigger area, you can find out how the entire big patch, you know, there is a big patch was there, where? Pacific Ocean, correct? So that big patch is moving, and people are targeting, people are tracking that. Lot of research is also happening. Now I think this is all about that. Now we will talk about Indian context. What is India can do. Now I will show you a picture of, you know, Google images of our own area, which is very, I think 40 kilometers from here. There is a lake, it is a confined lake, it is very static lake, where you can see all the plastic wraps are visible, correct? Now we went there and we did some experiments there, whether our systems, whether the spectral signatures what we collected from that locality, whether it is possible to, from a satellite image, can we get this type of pictures. But this is a very high resolution picture. Nimalan, can you tell something? Yeah.
So yes, so here as sir suggested, this is the Google Earth engine of a plastic raft which was artificially floated in a lake that is called Kolavai Lake, which is situated like around 40 kilometers. So what we did is, like, we tried to get the satellite image of the lake, and we tried to apply the true color composite, false color composite, and different indices that can be potentially utilized for detecting a target. So from the FCC of different images, so IRS list 3 could not be in a position to get the target, which is due to the lesser spatial resolution. But wherein if you see the list 4 image, there you can get the target which was there in the rounded, that circle part. So when we see the hyperspectral image which was called as high C, there you could not see the image, though we have a hyperspectral image, but the spatial resolution of the image is very coarser, so we could not get the targets. So this is like the potential of Indian remote sensing, like available satellite sensors in terms of detecting a particular target. So the inference is like, though we have a 64 band of information from the high C, due to the coarse resolution, we could not get the target. So the message is like, though you have a higher spectral resolution, you should have a very good spatial resolution to detect a target. Then when it comes to list 3, the same case, like you have a coarse resolution, but then you have only 4 bands. That is the reason you could not get any target in the second image. But in the third image, though you have only 4 bands, but the spatial resolution is very high, that is 5.8 meter. So you could get the target at least in the, like here you can see. So in the image 3, list 4, at least it is trying to detect something is there in the image. So therefore, for the litter monitoring, both spatial and spectral resolution plays a crucial role. So for better spatial resolution, one can always rely on drone images, but the thing is, like, we cannot always go and fly, and rather depends on whatever is available for free. So this is like the output. So when you take high C, you cannot detect this thing, and you cannot classify the image because you don't have any better spatial resolution. But when you go to list 3, you can detect the target, but you cannot classify it. But in the list 4, you can detect and you can classify like which is litter and which is non-litter pixels. So yes, you can tell about the spectral reflectors. So yes, in continuous to that, sir told like significance of spectral characteristics. So what NCCR, what we did is like we took 2 beaches of Chennai. Most of you must be aware of Besant Nagar Beach and Marina Beach, which is very famous for their tourist spots. So what we did is we tried to understand what is the spectral behavior of litter type and non-litter type. So when we go to beach, we tend to see different types of litters. So litter not only per se non-degradable litter, we can call degradable things also as a litter. So whatever they throw on the beach, we try to accumulate everything in a place and we try to get their spectral behavior. So as you can see these things, so the first thing is like a biodegradable litter target spectral behavior, and the second column is like non-biodegradable litter targets. So the major thing when it comes to degradable thing is you will get an absorption around 575 meter, which is a chlorophyll absorption, but which is not there in the non-biodegradable target. So that itself is like you can classify degradable from the non-biodegradable. But then we are trying to infer some different things, like how plastics can be differentiated from wood, how glasses can be differentiated from wood, how stray foams can be differentiated from wood. So those kind of inferences we are trying to get. So when we see the output, for instance, the plastic targets has a peak around 525 to 550 nanometer region, which is not there in the other targets. So likewise, when we see a degradable target, it has a absorption around 650 to 675, which is due to chlorophyll absorption, which is not there in the non-biodegradable targets. So again, same way when we go to the 375 to 525 region, so there you get a better distinguished pattern of spectral behavior when compared to degradable and non-biodegradable. So the message is like, on the preliminary level, one should try and attempt to differentiate the biodegradable and non-biodegradable. Then we can go and try with wood, how glass can be separated, how stray foam can be separated. Then we should get into how different plastic targets can be separated among each other. So yes, though we saw like different advantages of the remote sensing, still we have limitations. The major limitation is data scarcity. So ground truth data for marine litter is not so often available. Then sensors limitations. As I suggested, though we have a hyperspectral sensor, but coarser resolution restricts it from detecting a particular target. But on the other hand, though we have a list 4 with very good spatial resolution of 5.8 meter, but only 4 bands will not provide any meaningful outputs. Then when it comes to litter, we have a mixed pixel challenges. Like Dr. Dash said, you cannot get a litter like pure litter in particular pixels. You will be getting 4-5 litters together, then distinguishing among them or separating among them will be a major task. Then when it comes to environmental inferences, litter will not be like as such. It will mingle with water and there are lighting conditions, and most of the litter will not be floating. Some might get into the water. So there are certain limitations in terms of remote sensing. So these are all like summary of readily available spectral data sets. Like I said that NCCR developed a spectral database for different litters. So here you will get a global data sets of different type of litters, which already people have done. Sir, you can take a snap. Yeah.
This actually you can take this snap.
So one this thing is like we tried to download some of the data sets of global library and we tried to compare our spectral signatures that what we recorded in the local region. So we got to know that irrespective of the location, spectral behavior of particular target is exhibiting a similar spectral behavior. So that is like, though it is a known fact, we verified with some of the litters and we tend to like confirm like irrespective of the global locations, the spectral behavior of particular litter target is same irrespective of their locations. So this is
so as of now, I think that okay, remote sensing, whether it is satellite, sensor or drone, anything, it can classify, it can give you the information. Then after that, next what? How long we will continue like this? I understood that the basic research involved lot of spectral libraries. Okay, that is slowly we are developing. Whole community is developing that. But ultimately, where we are heading. So that's why we have very clear-cut direction that most important, as you said, that sensor data sets, particularly marine litter point of view, a high spatial and spectral resolution data set is required. So that will come in the future. So data accessibility, then real-time monitoring, and AI integration. Artificial intelligence, only the hope I think we can depend on that, because in that at least you can put as much number of input data to that model. Then at least artificial intelligence, because it is nothing but your classification of images, playing with images. So the way artificial intelligence is coming now, I think you can try different type of models, what is available AI models, and you see that which can give you the best result with the different combinations. It is not like single one target, you with the different combinations. And for that what is required? I think Indo-Australia platform like this is, I think, can contribute a lot, where we can exchange our knowledge and we can exchange our methodology. So that's why when we have collected, I think, how many liters, Gayatri?
173 targets and 1600 observations.
So 163 target and 1600 observations. When we did that, we thought whether whatever we have collected, whether is it right or wrong. So we need to compare with similar type of global data sets. Now the data sets are well ready. Now we are thinking how to take forward all these things, because India, you will feel in India now, in any coast, you will find that whatever observation we have done only on the land, not in the water. So we have not gone. So our next target is that we will make experiments on the water by taking different type of liters and collect their spectral signatures, and after that we can come to a consensus. So I think this collaborations is very much required. This will be very much helpful for all of us to educate to educate this type of research to carry forward in future. Now I will with this I am stop here, but still I will tell you that my Dr. Maya will explain that when you go to the field, students might be thinking that when I will go to the field, where I will start, what type of equipment I will take, what type of measurement I will, how I will make, you might be thinking, no, where I will start. I think he will give a real-time feeling. I think he will show what we have collected during field time. I think he will show some 2-3 slides. Can you go ahead? Yeah. Because when we are thinking actually this master class, we supposed to teach you that how you can do, but time constraint and infrastructure constraint we are unable to tell you. Otherwise you write it down everything. Data is I gave you the link. It is freely available. You can go and download. We will tell you what type of software is available. So that software also all are open source software, and that you can download on your computer and you can try. Okay, so that type of effort you can do. End of this class, you can contact all of us. Okay, so during tea time, so that we will tell you the exact link where the software is available. So you try your own images and you see that how it looks like. Okay.
I have a quick question.
Yeah.
That link for satellites, so what's the temporal resolution of the image?
I'm talking about litter to environment. Is it comes overnight? It disappear overnight? No. Litter comes, litter accumulated, it is takes some time. Correct? So your revisit period of your satellite sensors should be defined in that aspect. Suppose I will tell you that one upwelling, one cyclone is crossing, or a cyclone is crossing, you will get entire coastal water will be flooded, and you know that immediate after cyclone there will be lot of debris will be accumulated in northern part of this beach. You know this. This is a regular phenomena is there. So what you want to do? You need analyze particularly that time how the litter is going. So you need every alternate day or every day also if required. But with a subject to high resolution. Ocean color sensors are available every alternate day. Of the same location it will take you the visit with 360 meter resolution. But 360 meter resolution you can get a patch, not a specific target. Yeah.
Revisit time for list 4 is 5 days and 3 is 24 days. Shall I so thank you, sir, and very good morning to everyone. And I'll thank Dr. Dash sir and Nimalan because they made my presentation very easy. They said all the basic information about the wavelength, reflectance and other stuffs. So I just directly go what we have done for this particular work. So the main objective of this work is we need to get the spectral signature for each marine litter. So what we have done is we have went to 2 different beaches over in Chennai, and we find the various type of litters, and we have collected the spectral signature for the litter. So I'll tell you this is for only the land surface. I'm not going to talk about anything on the water surface because we have used a spectrometer, ASD spectrometer. So that will be a wavelength and the range is between 350 to 1075 micrometer. So it is between that. So we can capture only the visible range and the NIR range. So if you need to capture the water body and other stuffs, we need a sphere range. So we targeted only the litter on the land surface. So why we have started doing this is as of now we don't know how the marine spectral signatures will behave in the field surface, because we have a lot of satellite images. There we can find the spectral signatures for the multiple things, but in the real time environment in the field, how the spectral signatures will vary from one litter to another litter, we don't know. So we started that. And second thing is nowadays as Dr. Dash said, like AML and machine learning are ruling the world in all the domains. So for that also we thought of applying the AML and artificial intelligence in the detecting the marine litter. So for that mainly we need training sets. So for the training sets also we can use this. For example, as you all know, maybe you all are working on the machine learning and artificial intelligence. So for that we need a training sets. For example, you will train, you take a fruit shop, and for example you take a fruit shop, and then you will have a many type of fruits will be there, but you don't know what is that what fruit. So first to identify that, what we need to do is first we need to label this is apple, this is orange, and this is pineapple. So once we have these kind of data sets, then you give something. So then we can say this is a label, this is a apple, and this is the banana. So this is the basic concepts of machine learning and artificial intelligence. So like that same method we are going to impose here also. So for that the unique ID is spectral signature. So in machine learning, we need a spectral signature to identify the object from the satellite image. So for that, we started this work. And these are the objectives. So first we need to collect the marine spectral signatures for the different marine litters, and then we need to do the classification. So for that classification, I said, so we will use a machine learning and artificial intelligence. So because this is the recent trend, and with that only we can't go with that. So for that we need to develop a database. So for that we developed a Postgres open source database. And then once we have all the data in the database, we fetch from database and we plotted in the dashboard. So this everything we did it in the open source platform. And as Nimalan said, like we have chosen Marina Beach and Bessener Beach because these two beaches are very famous, and we can get more litter on these two beaches in every weekend and Monday morning. So such a way we planned, and one fine Monday morning we went and we collected all the marine litter in these two different locations. And this is the instrument what we have used. This is a ASD spectroradiometer, and these are the these are the specification for this instrument. So as I already said, this will be a range between 325 to 1075. So this will capture the near visible and near infrared region. So whatever we can take in between this region, we can take this. And also this is without GPS. And second thing, each and everything we can't take it the litter thing we can't take a location, and then we can't go and search it in the satellite image, because there is a past time will be there. So when that sensor pass, and the dates and time will be vary. So getting a GPS location and finding that exact place the litter in the satellite image is immaterial. So we didn't use any GPS coordinates. And this is the methodology what we have done. So what we have done is we went to the site and we have collected the spectral signatures of each marine litter. And then second thing is we have taken the same litter into the lab condition also. So what difference will make us that real sunlight, how the spectral signature varies, and that lab condition, that lab condition light, how that spectral signature vary. So that thing also we need to check. So we have done that site condition and lab condition. And third thing is what we have tried to do is as we are working on the marine domain, so we thought to take that same the same litter and we float it on the water, and we can try to get the spectral signature. But the third one didn't work because our wavelength range is between 350 to 1075. So if we need to go on the water, we need a sphere band. So the sphere band didn't match with that instrument what we have used. So these things basically we know, but we start off trying when we go to the field and we trying, we find we can't do all these things because of this water range is different. So this practically also we have done it, and we got to know. And these are the some samples where we have collected. So we have collected plastics as well as degradable and non-degradable materials also. So these are some sample things. And these are some curves. So as already Nimalan explained all these things in a detailed manner, so I will go ahead. So the field photographs, and these are the field crew. So we went and we collected litter for at two different beaches. And so as I already said, so we have imported all this data into the Postgres database, and then what we have done is we created a dashboard wherein we can visualize all this data. So the first single data, what it will show is it will show the multiple litter comparison. So you can compare the multiple litter curve, how it is varying from one to another. And that second one is a comparison data. So we have taken a field condition as well as a lab condition, so we can analyze how the spectral behavior between in field as well as the lab condition. So these are the two different dashboard pages we have created by using R and its packages. So there is a package called Shiny, so we use the Shiny package, and with that we can do all these things without server. So it's a very important thing. So without server we have done this thing. So in this we have used the R and packages, and for database we have used the PostgreSQL. So for this we didn't use any server, so that's the main important thing. And also this is open source software, so we can use it. Anyone can use it, and you can get that. So this one hands-on training we can't make it here due to the IP issue. So if anyone want to interested, we can go to the main building, and we can see all this thing in the real time. So thank you.
Thank you, Maya. Any questions? Yes, sir.
That's great work in quantifying the spectral signatures of many different potential litter objects. I'm curious about the variability within a particular type of object, a water bottle, across similar objects in that class, many water bottles, and what that distribution looks like, and how that may or may not be statistically different from sand, beach, rock, those natural environments, because of course single spectra won't work with the AI models, and the AI model is only as good as all the data you're putting into it.
What we have done is we have taken a different type of bottles, and we have taken with sand and without sand also. So we have a different curves for with and without sand also. So that will go to our model. So as we all know, yesterday in Dr. Usha ma'am presentation also, she was showing that for the cardboard box, she has a different varieties of cardboard box, and we need to train our model. So as we are working in machine learning, so as much as how much quality training sets you are giving, that much accurate model and result accuracy you will get it. So that's what we have taken all the with and without bottles also. So in that try with water and without water. The water bottle. Sands and everything also we have tried, and we have keep it.
Can I add something? Good morning, sir. So we have tried to replicate the real-time conditions like the crushed bottles, the bottles half filled with water or say any drinks, and the bottles partially filled with the sand. So we have tried to record how it behaves, like capture the spectral information in the beach as well as in the laboratory condition. So as you mentioned, that's one of the challenges that we are trying to address, because when you see spatially, it is very difficult to categorize the type of litter. You can easily categorize litter and non-litter, but when you know the things are leveling up, and I'm interested in to know the type of litter or say the type of plastic, we need more and more information how the spectral behavior of each and every plastic, and not only type but also color also matters a lot. So this is our one of the motto to initiate this work.
That's great to hear. I'm trying to get a better sense of the scale of that challenge. Are we 20% there, or 2% there, or 80% there, and how dependent is it on number of images? It's a massive amount of work.
Litter to get this spectral signatures. And second thing is that's what we have selected 2 different beaches so that we can get multiple. So the starting stage, so we get something, we will get more signatures, and we will add it in our. And second thing, with this we are trying.
Sorry, I have another question. I've been told not to ask about accuracy, yet I'm going to ask. Primarily just trying to understand, maybe not from your direct studies, but maybe you are aware, because I'm not, in terms of the improvement in quality of those models from just identifying litter on a beach or floating using just normal machine learning AI approaches versus actually having those signatures. So going through all of this effort, how much of the actual improvement to modeling and mapping marine litter you expect to achieve?
The nearby you might have seen that dump yard. So in that dump yard, finding a type-wise litter in the satellite image is immaterial. So what we are planning to do is we will find out the mixture of the literature, spectral signature. So once with that we will start. Then we will refine our model to go with the type-wise things. Coming to the accuracy part, we also can't tell as of now because we need to work, because as of now we developed 2-3 models. So one model will giving a 70% and another the same thing we will apply for some other images. So that will give a 65 to 70. So it's basically depends on the training sets and then what algorithms you are using. So for example, we have a lot of algorithm like random forest SVM. So with this itself we are getting the same training sets we are using, but the model we are different model we are using. There also we are getting a different accuracy things. So as of now we can't say this much we are expecting, but we will work and we will get and we will.
Now, to add to Maya's point, the first thing is like when you say accuracy, we followed a standard protocols that are there to measure a spectral signature of particular target. Like you have to maintain like particular angle to get the readings and everything has been followed. Then what we did is once we collected the data, we compared the data with the reference data. Like say for example, you have a plastic signature, you have a stray foam signature, and everything has been already recorded by someone else and kept in a database. So that is the main reason why we did the comparison part. So when we try to compare with like 3-4 different regions, like where, the existing study, so we were convinced that the whatever spectral signature that we are getting in this region for a particular target is in line with the targets that have been already recorded. So that was one particular this thing. Then when it comes to utilizing the spectral signature for classification of satellite imageries, that is why I said rather than going for a finer level classification, initially we are trying to understand how well the different materials can be distinguished. Like preliminarily we need to distinguish wood from glass. That itself is a bigger achievement. So then only we need to go for based on, like if we have a better spatial resolution, then we can go for finer things. But we'll get a better resolution in terms of drone, but the limitation in drone is we'll not get a better spectral resolution. So it is like you will get advantage and disadvantage in both the things. So we need to try and understand on the preliminary stage, detection is fine. So everyone is convinced that remote sensing can detect a litter, and to a certain extent it can track also. But when it comes to like further level, then we need to work on, that is why like when we are preparing a vision document for 2047, we recommended that we have a specific satellite sensors for multiple purposes. But then we have never addressed these kind of issues. So we suggested that we need a specific sensor with all this information, like with the available spectral information, we can place spectral bands. So that is what the purpose of collecting spectral signature is like. Like you know where and all you need to put a wave bands. So once you have that information, then we can always recommend the government or the people who are working in developing sensors and ask them to do so. So it is in infant stage, maybe hopefully we'll get better and better as we move forward. Yeah.
addition to that, I would like to add that, the future, actually, even I also believe the future of remote sensing, if you talk, it is the hyperspectral with a high resolution. It will, because our demand is not there. So, but if you get a high spectral, high spatial resolution of hyperspectral sensors, I think any type of litter, even dump yard also, it can tell you that what is non-biodegradable and what is biodegradable. That itself is the references. Because if you quantify that, if you take one picture and if you get what is biodegradable, what is non-biodegradable, then I think that will solve your problems.
Maybe that can be the last question. We are running out of time.
Mine will be quick.
Now there is online.
And there is one online question as well. Maybe after Marina, we
No,
can.
that's
Dr.
okay.
Max.
I can catch up at tea time.
Yeah. First, I
Let's
will
go for the online.
Can we get the details of tools as we are attending online? It will be very useful if it is shared by Dr. Jay Bhavan.
Can you hear me?
Please share the techniques. We will share with you all the tools and techniques. I think data already we have shared with you. This PowerPoint is also will be shared with you. So please contact with us. No problem. Thank you.
Am I audible, sir?
Yes.
Sir, actually, I have one question for Dr. Mayan, actually. Another question.
Yes, please.
Sir, actually, you worked on spectral signatures.
Yep.
So, have you tried finding some spectral indices based upon the spectral values that you have obtained?
That's what I said, ma'am. So we are working on it with the remote sensing also. So we will come up with some result soon.
Sir,
So
yes,
indices, sorry?
Although you are identifying spectral values for individual items.
Yes. That's what I'm saying.
But mostly the litter on the water surface, it is going to be like a patch.
No, that's what I said. We are not working on the water. We are working on the land surface. And then we are not...
Land surface?
Yeah.
Okay, sir.
So as I said in my presentation starting itself, we are working on the land surface that is in the beach.
Okay, sir.
So we collected
My mistake,
we collected the data also for the beach area only. So that wavelength also will be on the land surface only, not on the water.
Okay, sir. Thank you, sir.
Yeah.
Okay. Thank you. Thank you, sir. And thank you, Dr. Maya and Nimilan, for the technically rich and methodologically rigorous session. Now, as a token of our heartfelt thanks, I kindly request Dr. S.R. Marigauta to felicitate Dr. Das. And I also asked Dr. S.R. Marigauta, sir, to felicitate Dr. Maya and Nimilan as a token of appreciation. Next to Dr. Maya. Thank you. Next to Mr. Nimilan. Thank you, Dr. Dhanapalan, sir. Thank you. We shall now break for tea and reconvene here at 11:30 for the continuation of this master class. Thank you.
good morning. It's Anita. Thanks for everything, also for the help by email.
Yeah, I hope we can have you next time here. We have
Yeah,
another
me too.
Yeah.
It was a long trip, and this time it couldn't. Thanks again for everything.
Okay. So I'll
Do
be
you hear
back.
me?
Yeah, we'll try to be back in 5 minutes because it's a break. So I'll try to come back.
Okay.
Yeah. Thank
Do
you.
you hear me well? Do you hear me well?
Sorry?
Do you hear me well?
I didn't get you.
How is the speaking? Do you hear me well?
Yeah, I can hear you well. Your audio is clear, and maybe
Continue the discussion on spatial mapping techniques, expanding into multi-sensor data integration, algorithm development, and case study applications in marine debris reduction and tracking. First, he will be talking about the marine litter surveys on beaches, traditional method, European program, and novel global insights. And he will be briefing about the use of drones for marine litter surveys on beaches, techniques, and advances. It is my pleasure to welcome Dr. Umberto, a distinguished expert in coastal processes and environmental monitoring. His research harnesses the power of remote sensing to explore beach dune morphodynamics and marine litter pollution, providing valuable insights for advancing sustainable coastal management practices. Over to you, sir.
Yeah, good morning, everybody. I hope you can hear me well.
Yes, sir.
Okay. I will try to share my screen. Meanwhile, thanks again for the invitation, and it is a pleasure and an honor to be part of this workshop. I will try now to share my screen. Can you see the presentation?
Yes, sir, we can see. Can you please put it in a presentation mode?
It is in presentation mode already?
Yes, yes. Yeah, thank you.
Okay. Okay, good morning, everybody. I'm Umberto Andriolo. I'm a postdoc at the University of Coimbra and UNESCO Coimbra in Portugal. And my main activities are focused on the use of close-range remote sensing for studying coastal morphodynamics and marine litter pollution. Today, I will talk about marine litter surveys on beaches, about the traditional method and the European program. This will be just a brief introduction. And then I will talk more in depth about the use of drones for beach litter monitoring, and about the technique and the advances that this platform can provide, especially about the integration of drone-based litter surveys, beach dune morphology, and inshore hydrodynamics. Regarding marine litter or marine waste, we know that is defined as any item lost in the marine environment. Most of litter generates on land, enters the ocean, through rivers, and eventually part of the floating litter strand on beaches. Litter is a global issue that can harm ecosystems, marine life, and human health. So there is the need of monitoring litter to identify the sources, to check the trend of abundance, and understand the pathways. Regarding the definitions, we focus today about macro litter items, which are I got some echo, which are defined as the items that are measure of 2.5 centimeters on the longest side. So on beaches, the traditional methods rely on visual sensor surveys. Here, items are categorized and recollected generally over a 100-meter long shore transit between the shoreline and the back beach limit. And often, these are based on voluntary activities. In Europe, the OSPAR monitoring program started and ran from 2002 to 2020. About 236 beaches in 9 countries have been monitored. These surveys run basically one per season over the 19 years. There were some areas defined depending on the water body or on the coast. And you can find the details about the surveys that are available online on Excel spreadsheet. This program had the main advantage of defining.
Eric, can you switch up your mic?
Sorry?
Eric, can you switch up your mic?
What? Why?
You can, no, not you, Eric.
Okay.
Yeah. Yeah, Dr. Eric, switch off your mic, please.
Sir, it is off only, sir. It's not on.
Showing somewhere near, yeah, you can check.
No, sir, it's all off. Thank you.
Yeah, Dr. Ambarto, you can continue.
Thank you. So I was saying about this program, the details can be found in the guideline for monitoring marine litter on beaches in the OSPAR maritime area, which is this guideline that define first the methodology to survey and to standardize the surveys and to promote the comparison among the data collected on beaches. The first definition was the area to be monitored. So the area was defined as the 100-meter transect along shore and between the shoreline and the back beach limit. There was also a second survey to be conducted along 1 kilometer just for megaliter. So today I will focus about the macro litter to be monitored in the 100-meter sector. So the people goes on the beach. They provide also a kind of report to be a kind of spreadsheet to be filled with the type of litter that was encountered, and finally they recollect the items. The guideline also set a list of about 136 items. This list is nowadays a bit longer. The categories, the main categories are based on materials. So we had plastic, rubber, cloth, paper, processed wood, metal, glass, sanitary waste, medical waste, feces, and other unidentifiable items. This is an example of the questionnaire that must be filled before and during the surveys. For instance, this just define the location and the type of beach with other details regarding a first overview of the beach. And here you have an example of the list that is provided with some photo and example of the items plus the translation for each item in the different languages that of the countries that are participating and were participating to the program. And finally, the list with the OSPAR ID label that characterize the item. The main results of the OSPAR program between 2018 and 2020, but many other reports are also available. So the litter level on average was 252 items per 100 meters of beach. Plastic dominates with about 90% of the litter bulk. Nearly half of the litter consisted of single-use plastics and maritime-related items such as fishing gear and packaging. From 2015 to 2020, most of regions, except the Arctic, showed small decreases in litter levels, about 9 and 12 items per year. However, there is quite high regional variation. For instance, the Bay of Biscay, that is north of Spain, had the highest litter density, while the wider Atlantic had the lowest. And overall, the major sources were individuated in the sea-based activities, such as fishing, recreational use, and sewage-related waste, like cotton bud sticks. also, a large proportion of litter items are unidentifiable plastic fragments, especially on the North Atlantic coasts. Therefore, this complicates the source tracking and the cleanup effort. And overall, the program, apart individuating the most common items on the European beaches, also supported the policies to ban some single-use plastic items, such as cotton bud sticks in some of the countries. Apart monitoring, we know that we also need to improve the cleanup. Because regular, organized, and optimized cleanup activities are quite rare on littorals worldwide. And we need to improve the knowledge on litter dynamics to promote new cleanup activities. Lately, we review in this paper that you can find here, and during my presentation, you will see the references here below. We review 80 publications reporting the type, abundance, and weight of marine litter between 2000 and 2024. In particular, there are 7 publications from India and 4 from Oceania. And we analyze this.
Dr. Ambertu, can you please share
can you
your
hear me?
screen? No, the content sharing has ended.
Okay, sorry. See. Okay, can you hear me?
We can hear you, but we couldn't see your screen.
Now?
No, we couldn't see.
Okay. Okay.
Yes, now we can see.
So I was saying that we reviewed the state of art and literature because we wanted to investigate the weight of marine litter. This kind of parameter is quite rare to be read in literature because the weighting task is kind of difficult in the field because you need the portable scale or you need the laboratory. So we wanted to understand how much does a marine litter item weight on the beach. And we also wanted to see what is the plastic percentage based on continents and water body. So what we found is that overall, a litter item weight on the median value of weight of a beach litter item is about 13 grams, while the plastic item weight about 9 grams. We also, you can see here how the median weight from Europe is 8.6, then the Oceania was found to be much higher, while on coast, depending on the water body, on the Atlantic, the median weight was about 9.2, and the Pacific was almost the double. However, you can see that overall, the median weight of litter was consistent over the continents and the water bodies. Plastic, instead, was quite more variable, and but also comprised between these values. And then what we found that the plastic percentage was comprised also between 60% and 86% around in the Atlantic Ocean. However, by weight, if we consider just the weight of plastic in the litter bulk, this doesn't go up higher than about 60% with the median value of 50%. This is quite important because for organizing cleanup activities, we can estimate the weight just considering the number of items and the percentage of plastic. I have finished now the presentation about this introduction. So if you agree, I can go on with the presentation regarding the use of drone for beach litter monitoring, technique and advances. Also, in this presentation, I will show just our works, because we had investigated most of the aspect of the use of drones for beach litter monitoring. Our works are among the most cited in literature. Obviously, you can find many other, not so many, but other works in literature. So I invite you to check also the references inside our works and to explore this technique. As we saw, the traditional monitoring programs rely on visual sensor surveys. However, these kind of surveys are human effort demanding and are very limited both in space and time. So the question was, can we exploit aerial images for marine litter monitoring on beaches? The first pioneering work regarding the use of aerial images come from Japan in the 2011. They just used a camera, digital camera that was attached to a balloon that was left in the air collecting aerial images. Here, the items were, I'm sorry, but I have someone speaking in the mic, okay? The items have been identified and the accumulation were identified by these aerial images. So from here, it was the first example to use aerial images to map and to check the abundance of litter items. We know that in the last 2 decades, drones came to the market, and they are quite I'm sorry, but they are quite versatile for the environmental monitoring. So we know that they are lightweight to transport, deploying remote areas. They provide high resolution imaging because they are equipped with HD cameras. They can fly autonomously, following a pre-programmed flight path and using GPS and reducing, therefore, the manual operation. They can also be equipped with gyroscope and GPS for stable, accurate hovering and movements. They can also provide real-time data transmission and integrate different sensors. But today I want to talk about these last two aspects. So in the environmental monitoring task, they are cost-effective because they are more affordable, for instance, than satellite imagery or manned aircraft. They reduce the environmental impact because they have minimal disturbance to wildlife and ecosystems compared to ground-based or manned methods. They also allow a fast data collection because the deployment is quite rapid and the coverage, even if it depends on the battery autonomies, can cover large areas in a short time. Reduce also the working load and improve safety because it eliminates the need for human effort to enter in hazardous environment, and also allow a high frequency because the time-sensitive data in both localized studies and large-scale environment surveys can be frequent. And also, in the end, it enhances the decision-making because it is accurate and up-to-date information and provide up-to-date information to support the better environment. In the marine environment, there are many applications of drones. For instance, it allows the 3D reconstruction of coastal features such as beaches, dune, and cliff morphology. Can be used to characterize the vegetation and the mapping of the, for instance, dune environment and cliffs. It has been used for marine wildlife monitoring, such as the mammals and birds. Can also be used to check the temperature, the presence of sargassum in marine waters, and also to monitoring the marine disasters such as oil spills. Many other applications can be found in the different reviews that can be found in the state of art. So from here, we say, why not use drones for litter monitoring? Here you have the pictures of some of the drones that we tested and we had at the University of Coimbra and ITNESC Coimbra. So this is the DJI Phantom 4 Pro, the RTK, and then the DJI Matrix 2010 that has double gimbal, so it allows us to mount the X7 RGB camera, that is a high quality camera, but also the multispectral Altum Micasense, which has 5 bands. Beside the RGB camera, the RGB bands, it has also the red edge and near. Here now I show you the 2 study sites that we choose. So the first beach dune system where we tested the techniques is at Leirosa. We are in the central coast of Portugal, between Lisbon and Porto. This area is composed, it is a beach dune system, meaning that it has this beautiful open beach facing the Atlantic Ocean, and the dune ridge and the back dune, which is a vegetated dune, quite stable, with the fore dune and the back beach. It is this particular location is downside the estuary. So we have an estuary here of the Tago River, and it is a mesotidal beach, meaning that the range is about 2 meters between low tide and high tide. And it is an high energy coast because the mean itself is about 2 meters, while the storms can reach about 5-6 meter high wave. Also the wind is about 5 meters per second, but the wind gust can reach normally during winter also 50 and 60 meters per hour. This is a picture taken from the beach. You can see accumulation of litter and the dunes that are in this particular picture and part of the beach are under erosion. The second study site is also a beach dune system, which is Chiaios. Is very close to Lei Rosa. It is north the estuary. It is also a natural, well-conserved dune ridge, which is about 20 kilometers long. The coast is the same. You can see, for instance, here, on that day, it was about 3 meters wave high. Also here, you can see that the dunes is very well developed. We have here very natural dune ridges. So we have the embryo dunes that forms here before the foredune. The foredune is vegetated with herbaceous, mostly herbaceous vegetation. And then the back dune, where you have more shrubs and more stable vegetation. Here, the foredune is about is between 6 and 10 meters high. The experiences I will talk about were done within the WASP for Litter project. The PI was the professor Gil Gonçalves, who prepared also the presentation with me. So at the start, we tried to set up a kind of protocol to be followed to finally map and the marine litter presence on beach and dune. The experiences that I will talk about started then to think and how deploy the drone on the beaches and dune. After that, we apply the structure for motion photogrammetry to processing the aerial images. We explored both manual image screening and machine learning to detect the litter items on the beach, on the image. And then we also investigated how integrate the environmental forcing and morphology to this to the map of litter, trying to understand the dynamics and the litter dynamics both on beaches and dune. And finally, we produced the marine litter maps in the way that I will show you later. So overall, this was the main idea, not only provide a census of items on the beach, but also trying to advance the knowledges on the beach and dune pollution. We also explored the other works, as I was saying before, because the main idea was to propose an homogenized framework so that the data collected by drones worldwide would be comparable and homogenized and standardized. We found in litter this review was from 2022. We found at time about 16 works, mostly in Europe. Nowadays, I think in the last 2 years, there are at least 3 or 4 more works, especially from the Baltic Sea. And here you can find the different colors regard the fact that this orange indicate just works that have been conducted. I'm always talking about drone-based litter surveys. The orange color regard the application on beach, while the green also considered dunes. The the use of drones, I will talk also later about this. The use of drones on dunes was also kind of original because the visual sensor service do not take in consideration dunes, and the survey of litter on dunes. First, because on dunes it is avoided and should be avoided to walk on dunes, and then it is much more difficult from the logistical point of view. Therefore, drones are particularly good for surveying the marine litter on dunes because it is the impact is basically zero from the ecosystem point of view. So we revised the different parameters that all other researchers used to fly and to analyze the images, and we also analyzed the assessment. So I invite you to read this paper. Historically, the first work was from Daydun here in Malta. It was the first appearing the use of drone to map the marine litter items. I believe on a very different sites. They used manual image screening, and they tried to replicate the OSPAR and survey using images. After that, there was Martin in the Red Sea, Arabic Sea, who also was the first to explore also the potentiality to map the marine litter on low-lying dunes. And after that, mostly the works were from 2020. Yeah, it was a bit weird to not find any works on the Pacific, and apart the Takaya from Japan, and also in the continent of America. Nobody used so far obviously from the scientific point of view. Revising all these works and based on our experiences, we end up drawing up this kind of framework in which we distinguished four different stages. The first stage regards the plan for field experience. The second one, the field deployment. The third one, the image processing and analysis. And the fourth one, the litter service assessment. This kind of framework is quite simplified. However, the four main tasks are here and are well defined. Today I will talk in details about all four stages, and I will try to share our experiences and a bit of the state of art, and also try to talk about the potentiality and what can be developed more to improve this drone-based litter surveys. So regarding the first stage is, as I was saying, the plan for field experience. We define we need to check the flight regulation and legislation, environmental conditions, and the topography of the study area. So first of all, it is necessary to check the national flight regulations, such as the need for patent to fly the drone, and to check the local flight regulation. There are often, for instance, in Portugal, the maps in which drone can be flown, also the altitude that is allowed, and some days are off, and so on. I can talk talk about Europe, the regulation are getting more strict, for instance, and so the first thing to do is to check this. After that, we need to check the environmental conditions for the day of the field experience. For instance, for the phantom that I showed you before, the wind threshold, but the wind must be lower than 20 kilometers per hour for flying. However, depending on the drone itself, it can be higher. For instance, the Matrix that I was showing, the big one, can fly with, I believe, till 20 kilometers per hour. It is better to choose, obviously, a day with clear weather. And we also learned that it is necessary a site inspection before the experience. Because first of all, it is necessary to check if there are obstacles to fly the drone, but also to check the beach profile, and so on. And obviously, you need to charge the drone batteries, because it happens obviously to everybody, I think, to forget that. Regarding the stage 2, that is the drone deployment, there are several parameters that must be set before flying. We did several experiences about this. You can find also in literature some other works that focused on this. First of all, it is necessary to set the flight altitude, the trajectory, and the speed in the drone software for the autonomous flight. Then you need to set up the camera, so the shutter speed and image overlaps and the ISO. And in our case, we also wanted to map the litter, so we also needed the ground control points to georeference the images. Here you can see the X. In this list, it is obviously much important. The most important is the flight altitude. And I will talk now in details about this, because the flight altitude is the main parameter that determine the ground sampling distance, which is the image resolution. The ground distance for the flight height, for the pixel size, divided for the focal length of the camera. So the focal length is the parameter of the camera and cannot be changed. A better camera is obviously a better focal length. The pixel size is what you will get, what it is determined also by the camera. Therefore, the flight height is the parameter that determine the image resolution. To detect marine litter on the images, we need a proper image resolution. Here you can find the differences about the ground sampling distance. For instance, you can see how the resolution degrades when the ground sampling distance augment and increase. We explore several heights with our drones, and we end up in this last publication. We end up defining a kind of a range of ground sampling distance that should be used for detecting marine litter on images. You can see we define the marine litter item, the minimum in 2.5 centimeters square, and also we explore what would be the image resolution to detect at least 5 centimeter. This because during our experiences and our colleagues' experiences, we found that the 2.5 centimeter, taking 2.5 centimeter as minimum size is quite difficult in the field. And we also found that these kind of fragments are difficult to be recognized and to be classified. Therefore, a more coarse, let's say like this, survey would can be taken also as 5 centimeter as the minimum size to be detected. So we define the number of pixels to detect this kind of these small items, and what we found is that a 0.5 ground sampling distance would be enough to detect at least the 2.5 centimeters, while a 2.5 centimeter pixel would be good to detect at least the 5 centimeter item. Here again, you can see the range that we define between 0.5 and 1.5 centimeter pixel. And we also tried to give some examples considering the drones, the most common drones that you can find in the market. Some, the cheapest, have a fixed camera, so it was quite easy, for instance, for the Phantom 4 Pro, to define the range of flight altitude. You can see that the Phantom, for the minimum GSD, should be flown about 18 meters, and for a coarser survey, it can be flown till about 42 meters. What also you can see in this graph is that the bigger drones that has a gimbal in which you can mount the camera that you prefer have a higher altitude, can be flown at a higher altitude, but also has a longest range in which you can decide the flight height. The flight altitude is also important because as higher you fly, to obtain the same ground sampling distance. If you fly higher, obviously you can cover a larger area in the same time and with a short time. This is because each drone has a battery autonomy that is still quite limited. For instance, the Phantom can fly for about 20 minutes, half an hour. And you understand that if I can fly higher, I can catch a longer area in the same time. So this kind of examples can be good to give a first perception of what a drone can. First, it set the minimum and the range of altitudes that must be chosen, and second, it can be seen that a different drone can cover a larger area. So once we set the flight altitude, the camera, and we float the drone and we collect the images. And the stage 3 regards the image processing and analysis. I want to talk today about the structure for motion to generate the orthophoto and the digital surface model. but I will focus more on the litter object detection on the photo. I will show you some examples both of manual image screening and automated detection. This you can see a first example that we get from the fly. I think this is from 20 meters fly height. The items can be recognized. We have in the coast of Portugal, we had many of these octopus traps can be found on the beach. These are items that are anchored on the bottom of the sea, released by the boat, and after a few days the boat come back to recollect them with the octopus inside. But there are a large part of these that are lost because of the turbulence and waves and so on. So you can find many of these items on the beach. This is processed wood. You can see some fragments, some plastic bottles, and fishing ropes. With the manual image screening, basically, you have this picture. We tested. You have the picture. We in QGIS, and we try to simplify the manual image screening using this kind of list. So indicating a code for the material and the code for the color, also including the non-identified or not listed item. So, for instance, we went on the QGIS. We marked the items, and we characterize the plastic bottle with the plastic and the transparent color, and also the number we found of this. We also explored what was the accuracy of the detection from different operators. This was was a work with about 13 people or more. I don't remember exactly the numbers. It was quite interesting because before the other authors just focused on the image resolution and the GSD to see how this influenced the detection of some items and the recognition of some items. Instead, we provide the same images to many people to see what was the influence of the influence of, for instance, operator expertise on marine litter, expertise on using image processing software, expertise in different disciplines. So we understood several things. For instance, not only the GSD determine the accuracy of the detection, but also on the operator patience and experience in marking the items in GIS tools or in using some softwares, on their own knowledge of the litter type, material, etc., which is important also for training machine learning, and also on the nationality and territoriality of the operator. For instance, we found that the colleagues from Italy didn't know some of the items that were present on the Portuguese beach, and the contrary, some Portuguese colleagues didn't know some items that the Italian colleagues found on the Italian beaches. Here we also proposed from this work a kind of homogenized list that of litter to mark the items on the beach, but this it is a longer talking that is also particular, so I want to go in details about this. Then we explored also machine learning techniques. We tried the pixel-based and object-based detections. What we found is that object-based seems more feasible. We are not actually experts on machine learning. Surely you can find better works, especially from the last 2 years, that explore exactly deep learning and machine learning with more expertise also. The main issue of machine learning is that you need many images to train the codes. There is a colleague from Greece that is exploring much more this kind of aspect with deep learning. What we wanted to try, it was especially the approach based on litter items colors detection, because we were seeing that based on colors, the algorithm was quite easy to be set, and then the items were quite visible also in the vegetation of dunes and on the beach. And most of the same items as often the same color. I mean, in our course, for instance, bottles are 90% transparent. We have many styrofoam pieces that are mostly yellow. The octopus pot that I was showing before, the octopus traps are 99% black. So these colors can help for easy setup and automatic detection. What we also investigated is the segmentation of images. This was quite interesting because the size of litter is another parameter that was missing, or it is missing, from the visual sensor surveys. This allowed us to segment the images and measure or estimate the size of items. Also allowed us to see and to derive some interesting parameters. Here you can see we could measure the length and the area and the ratio between length and width. So we found, for instance, that bottles are always about 15 centimeters, and then we found the wood was basically bigger than other items, and so on. And later I will show you also how you can use this kind of size analysis. Here are other examples of the image. You can see that the items are quite recognizable. Here you have the fishing gear, some pieces, fishing net, a piece of wood. This was probably undefined, and then again the octopus trap and the plastic bottle. What we did also, and this is another advantage of drones, is that you can map and geolocate the items. That's why we use the orthophoto. We can geolocate and the items and cross. Here you can find the differences of items on dunes and on the beach. Crossing all this map with the 3D model of the beach, we could also found where and how and why the items were more accumulating in this part and in the central part of the beach. And we'll show you later about this. Finally, beside testing the litter colors, we also tried the multispectral camera. In this work, we tested the spectral angle mapper classifier, which basically take the 4 bands, and it is a technique that usually is used for hyperspectral cameras, but we applied this on the multispectral. The technique basically tried to compare the response with different angles between the bands of single items, which in theory had different spectral responses. I was saying briefly that before the talking before was talking about the spectral responses. Unfortunately, I couldn't follow the whole section. Anyway, what we did was like placed some items as samples to sample the spectral responses on the beach. We collected most of them between the area that we were flying on, that was about 80 por 80 meters, and how the algorithm basically works. This, for instance, was a plastic bag. The algorithm checked the spectral responses, find the angles between the bands, and then we went on the area to see if the spectral angle mapper was finding another plastic bag. The test was quite easy to set up, and it was just to see how the multispectral cameras could be used operationally. Apart the automated detection, I have to say that you can read the details in this paper. The algorithm did work well. Apart that the area was quite complicated because there were several types of vegetations, several types of items. So overall, we were quite satisfied despite the low F-score. What we did also was to test the automated detection depending on material. So we found that the materials were quite well detected autonomously. Basically, as we know, 80% is composed by plastic in the little bulk. So we found that plastic was quite easy to be detected. Also among vegetation. Obviously, what we also understood is that the multispectral camera cannot, so far at least from our test, cannot distinguish type of plastics. It can be used in conjunction with the colors to at least estimate, for instance, the number of plastic bottles, transparent plastic bottles, crossing. These two, crossing multispectral and the colors, can be more efficient than simple multispectral camera and sample angle mapper. But it was quite interesting to test this kind of algorithm with the multispectral camera. Pretty recent work, because after we map the marine litter items on the beach, and reminding what for regarding the marine litter weight on the beach. We tested this kind of estimation of weight from WAV imagery. We tested 3 potential methodologies. So once you map all the items, if we found we tested, can we assign a weight to the number of items, to the size of items, and to the size of items, basically. What we found is that if we use, for instance, the median weight of a single item, we can also estimate the map of weights on the beach. This is obviously something that must be investigated more, but we wanted just to advance and to add another parameter to be estimated from WAVI imagery, just to close the whole possible delivery and assessment. I don't know how much time I have now yet. Sanit, if you can tell me, please.
Now, you have 20 more minutes, sir. Sorry. Now, can you hear me?
Sì, I can.
Yeah, you have 20 minutes more. 20.
20 minutes, okay. Do you want a break or I go on just to finish?
No, please continue. Yeah.
Okay. So, see, I don't know how many slides I have yet, but I think I can finish in 15 minutes.
Yeah,
I will show you now thanks. I will show you now the stage 4, so basically the litter assessments. The first obvious assessment is the characterization of the beach litter bulk. So we counted the objects. You can recognize the type, the material, and you can analyze the size of item, and as said before, also estimate the weight. And then what we provide also was the litter maps and the spatial distribution of items, such as the covered area, the density, and the hotspot. This here I show you the first work that we did in 2020. Basically, after this, we also, as I was saying before, integrated the 3D beach model. You can see here the height, the elevation of the dune ridge and the beach with the map. Here is the profile, and this is the heat map with the marine litter accumulation. In this case, we integrated the marine litter map with tide and run-up. So the beach run-up is basically the movement of the wave and the seepage movement on the waves on the beach and on the beach profile. So what we found here, for instance, is that first, the marine litter was accumulated over the highest tide and the 95% run-up, if I remember well. And it was accumulating where we had some beach depressions. Here, for instance, and here there were some depressions of the beach profile. So from this first experience, what we understood, for instance, that it was enough flying the drone just upper the mean tide, meaning that you can avoid flying to catch the area in the intertidal area. And then we understood also that it was important to see the slope of the beach, because some areas surely can accumulate marine debris and marine litter in the following days, depending on the beach depressions. This was the first experience. It was quite interesting also to see this kind of dynamics on the beach. When I was talking before, you have already seen the maps. Because we thought and we also how to show the litter maps. The authors before us just shown this kind of maps in which basically the litter items are mapped. You can see the different colors can be depending on the type, on the sources, or on the materials. But we wanted to integrate the marine litter location and spatial distribution with the beach and dune morphology. So we end up thinking on the grid generation, because if you generate the grid, then you can see better, for instance, the abundance. Here you count the number of items per cell, but then we can also see, for instance, just producing the thematic maps, such as the map of plastic, the area occupied by litter, and so on. When you cross this kind of grids with a 3D model, for instance, it's quite interesting to see how exactly the spatial distribution respond to the beach morphology, for instance. Also, this kind of grid facilitates the homogenized mapping. I don't remember, probably here we use a 4x4 grid, but then it is also easy to enlarge the size of the grid cell or downsize the grid cell. As I was saying, it is easier to integrate the different data, such as the digital surface model, the beach profile, and the run-up tide to describe the dynamics. It is specific, different maps can be produced and compared and coupled. We have seen that several authors after us try to start to use this grid, so we hope that soon we can find also how to compare the maps. This is again the same slide. I try to also to go a bit faster. Here are some pictures of the Lei Rosa that I was showing before. So we also wanted to, beside the marine litter, we also wanted to see how this dune ridge was eroding. Basically, it is just going landward, and it is under much erosion. So we end up with the idea of using the drone for multiple purpose. So we did 3 flights during before in the winter and then in the spring. So with the same flight, first we went to see how the dune was regrading and was eroded by several meters. You can see here that it was under erosion, while the beach here was a bit higher. And crossing this, we also went to map the marine litter on the beach. So this is the accumulation during the first flight, from the second flight and the third flight, plus the different items, how they were moving during between the different spatial distribution of different items during different flights. We crossed also this kind of map. We crossed the maps with the environmental forcing, such as tide, waves, and the total water level, and wind speed. What we have seen, and we could understand that, for instance, the octopus spots were easily buried by wind, while the plastic bottles, basically, they were moving southwards. Also, the wood didn't move much, and you can see that here the accumulation was due to the wave direction. Basically, you have this structure with this growing, so the wave diffraction waves were coming from the north, so the wave diffraction were accumulating the items downwards, the structure, while the tide is redistributed. After that, the tide and the wind redistributed the items on the south and on the upper beach. And after that, the wind gust replaced, removed all the items, most of items, over the beach. So this work was basically to see and to promote the multi-purpose flight of the drone. I will talk now later briefly about the dunes, what we found on the dunes. Here we set up the framework also for dunes, so it is similar to the one that I showed you before, but we check also during the field inspection with the mobile if we were well detecting the items on the images. And after that, we investigated, we estimated the size of the items, we mapped also the vegetation, and so on. The first work, we understood that items, here you have the density of litter, were accumulating in the back of the beach, on the shore of the foredune, where the main dune blowout, that are these interruptions of the dune ridge, were present. So basically the wind or overwash events were taking the items from the beach and pushing through these kind of interruptions. Also the wind was moving the items upper the foredune, and you can see here that many items were semi-buried by the vegetation, and I will show you later this. It was very interesting this because we were just crossing therefore the dune morphology with the maps of and the accumulation of items. We went further and we analyzed how the items were distributed over the cross shore. So we found that the embryo dunes, which are characterized by small plants, basically, were trapping most of our heavy items were found in the upper beach. On the foredune, we found that the herbaceous vegetation was trapping lighter items, such as the napkins and plastic bottles. And the shrubs in the back dune were instead trapping mostly styrofoam, which are pieces that are quite volatile, so the wind was transporting, and the shrubs were trapping this, and our octopus spot. You can go to see the distribution of recreational items, mostly that were on the foredune, and the fishing related items were mostly in the back dune. Also, we went north and see in the south. Basically, here you have a car park, and this part it was much more natural. So we found that the recreational items were closer to the car park, and on the north, more fishing related items were present. Here again, some examples, also how plastic and paper and wood was distributed, how the items were distributed over the cross-shore sections. So you can go really inside the distribution of items with this kind of analysis and with the data provided by drones. Here again, you can see that the percentage of plastic was how was distributed, the different items from the car closer to the car park and to more natural areas. And here you can see the back dune with some items trapped by the vegetations. And finally, during these surveys, we also observed that litter can be exhumed while zooming from the dunes. So this is not much related with drones, but I wanted to finish with these slides because it is quite, we were astonished by this. So this is Lei Rosa, is the last one, the beach with the growing. And you can see that we were observing many items that were coming and were exhuming from the dune under erosion. So we went to map this kind of event. What we discovered, talking with the locals, is that in the past the dunes were used as a dumping site. So it was a irresponsible dumping. There was much little attention at that time. We are talking about more than 30 years ago. And after that, the dune was nourished. Basically, they put, they brought sand and to stop the erosion, and they basically buried all these items. We were also kind of surprised because here we mapped the wall of the dunes, and we found items from the late 17. So basically, these items were from 81. So there were plastic items that are exhuming from 40 years ago. And okay, this was illegal dumping, but after this, we went also to see what was occurring in Chiaios. These are very natural dunes, and there is no intervention of any kind, any human intervention on this coast. And we started to see also items exhuming from this site. This was from Lei Rosa. You can see that also it was very important, the exhuming, but here it was just natural dunes. So we went to take pictures and map also here all the items that were exhuming. So you can see that these red boxes underline the presence of litters that were exhuming from here. Here you have the embryo dune, also the 4 dune wall. Here we are talking about 10 meters high, many items. So we went also to create this kind of spatial distribution maps. And you can see that, I don't remember the total number of items we counted in 2.3 kilometers, but there were hundreds. We start to observe, and we start also to think about how these items were within and embedded in the dune volume. So you can see that basically the embryo dune vegetation trap or stop the items. And then we found items within the roots of the eroded dune. Here again the embryo dune, and here again another other items exhuming from the eroded 4 dune. So we end up explaining the process that sink litter in the dune volume. And basically the litter participate in the embryo dunes growth erosion process. If you know a bit about coastal morphology, the embryo dune has the vegetation. If the embryo dune, in the embryo dune enough sand is trapped, it becomes 4 dune. The vegetation change, and the 4 dune just grow as a kind of hill. And so the litter is trapped by the embryo dune and by this vegetation. If the embryo dune becomes a foredune, litter items get buried and in the dune volume. After that, if the foredune is eroded by storm impact, the litter is re-exposed to the marine environment, what we were observing basically. If instead the embryo dune erosion, if the embryo dune doesn't grow but it gets eroded, and for instance the high tide wash up the items, the foredune, and there is also the other case in which the foredune grows and the litter is trapped by the vegetation. Therefore, it gets all the items get buried and trapped within the roots of the vegetation. This was quite astonishing to see this. So there is an urgency of improving and clean up on the beach, also because of this, also because the dunes contain much items and much plastic, which obviously can be dangerous for the vegetation and the ecosystem itself. So just to conclude, some take-home messages. Very briefly, we need to promote frequent drone flights on coast, also for multi-purpose studies. We need to integrate an integrated approach that use the litter surveys, integrate the coastal morphodynamics and environmental forcing to advance and understand the litter dynamics on coast, obviously to support the search for mitigation measures. We need an homogenized and standardized drone-based monitoring strategies. This is a picture from Tokyo, and we were working on a guideline that you can find at the Ministry of Environment of Japan. We try to. It is a guideline regarding remote sensing, in which also Mishra was working on. And there are many interesting observations and pages to work on this topic. And finally, we need to raise awareness on coastal and dune pollution among scientists of different disciplines, stakeholders and citizens, because we need to advance knowledge on litter pathways and to promote the cleanup. This last slide, just to acknowledge all colleagues from INESC, Coimbra, University of Coimbra, and who collaborated with the project, all the international partners who collaborated to the scientific.
Thank you. We will take a couple of questions from the audience. Thank you. We will take a couple of questions from the audience. Any questions? Yes, please. You can come in the front and speak in the mic.
Yeah, am I audible? That was a great session, sir. I just have one doubt. You are monitoring the types of litter. I just wanted to know, was there any collection of data about the kind of litter that was brought to the shore and things that were the litters that were already dropped on the shore? Because the transport of litter by marine routes is very high, and so have you found any data on the kinds of litter that were transported by sea?
Yes, obviously, in the multi-flight experience, we have seen, for instance, that there are some items that are easily resuspended because of the wind, while others were remaining on the beach because semi-buried. For instance, we have seen that plastic bottles were flying quite easily, also with very little wind, while some other items, such as the octopus spot that I mentioned before, were easily trapped by the vegetation, or they were semi-buried by the sand because the sand was entering inside that. I don't know if I exactly to your question.
I think
replying?
it depends mostly on the weight and the wind dynamics. But is there any specific data, as in like monitoring the waste? For example, after a tide, it could be different. So is there any possibility to map that?
Yes, of course. If what you wanted to do, it was to fly with high frequency. For that, it is a bit difficult because, for instance, in our coast, the days in which you can fly are not many, depending on due to the wind. Perhaps what you are saying is what, for instance, our colleagues from Japan are doing to try understanding the very dynamic in a very short term. It is necessary a continuous monitoring. They are mounting some cameras to monitor, even if the area is quite small. But it would be very interesting to see, as you were saying, the tide and waves, what are the main items that will be easily resuspended and some other that are instead remaining on the beach. They are collecting data now. We will see later what can be achieved or less, but I think that what you are saying, to see exactly the dynamics, it is necessary continuous monitoring of that.
Thank you, sir. Any other questions? Yes. Okay.
Thank you for your presentation. I think you are audible my voice. Thank you for your presentation. You are covered so many things: marine litters area coverage, and marine litters near to the beach. What is the weight of the pixel of the marine litters? Thank you for your great presentation. But my thing is, are you observed these marine litters near the coast in a hyperspectral measurement?
I have understood on the hyperspectral, but we didn't. Was your question if we tried? The question was regarding hyperspectral, right? Yeah, I know that some colleagues, you can check Balsi from Rome. They are trying, they are experimenting hyperspectral. We did not experiment it because the sensors are quite expensive and to be mounted on the drones. So we don't have hyperspectral camera. We tried just multispectral. Was this your question? We didn't, and I know just this works from Rome. I think the University of Sapienza, they are trying that. They are experimenting with the drone and hyperspectral. What we have seen is that obviously with the hyperspectral, you can try distinguishing also different kind of plastics. The main point is, because we also had problems with the multispectral cameras, is that how much is the sensor reliable in this kind, with this short distance, and how much is good the spectral response. In these terms, we have seen, okay, we tried a multispectral camera that is somehow cheap, but I haven't seen, we didn't try. I just read in literature about the multispectral, hyperspectral, sorry.
Thank you, sir. We have one more question from Dr. Marina.
Thank you, great presentations. I was just wondering,
We know that Europe is heavily invested on marine debris and how to tackle the problem, and it seems like drones could be a great technology to be applied to identify hotspots and monitor beaches, like you were mentioning. Can you explore if there is any appetite from the government and related stakeholders to use this tool to inform policy and make some change?
Sorry, the quality was not. Personally, we haven't talked to stakeholders. Apart, we proposed to the municipality a pilot study to explore the use of monitoring and to explore the drones to advance the knowledge. And because what we think is that you can use drones to surely increase the frequency of the monitoring and increase and advance the knowledges of dynamics. Once you know the dynamics, for instance, we observe that there are many accumulation points, recurrent accumulation points, while other sectors of the beach are not interested by accumulation. So focusing on that would increase and would improve the cleanup, for instance, operations that nowadays, at least in our study sites, are done just before and during, just during the tourist season. The second thing is that not only monitoring, but also we are we are trying to think on the use of drones also for cleanup, and we will see in the future if we are able to design a kind of framework to monitoring and clean up and monitoring and clean up and so on. For the rest, I have to say I have not talked with the policy makers or stakeholders. After 6 years, because we are talking of about 6 years that this kind of surveys have been done and have been set up. We hope that in the near future also the use of this kind of service can improve much also the policy and the ban of items, and especially for mitigation measures on the coast, it is necessary to advance from the point of view of litter dynamics knowledge. I don't know if I reply exactly to your question, but
Yes, you did. Thank you.
I hope.
Thank you. Okay, there is a message. Okay. No, that's not a question. It is for a thank you for delivering the speech. Okay.
Thank you to you all.
Yes. Thank you, Dr. Ambato, for that comprehensive overview of advanced remote sensing solutions for marine debris analysis. Thank you so much.
Thank you.
We now will break for lunch and shall resume at 2 o'clock. Thank you.
Thank you, Ambato.
Misha.
Thank you.
Yeah, I hope I'm not in the hall also.
Yeah.
Thank you, Mishra.
Yeah, thank you.
Nice to see you. Nice to see you. Next time, see you soon. Huh?
Yeah, we'll see.
Okay.
I will be in touch. Okay. Thank you.
Okay. Ciao, ciao.
All right. Okay. Please register. In the meantime, while you're registering, I just wanted to... Okay. This way? Better? Yeah? Okay. I'm going to be moving anyway, so... while you're registering, I just wanted to run a quick survey here in terms of what's the people's skills in the room, so we can basically account for that as well. So who here never used R in their life before? Yes. Okay, great. So we have a couple of people. Who here works with actually biological data on different species and anything like that? Okay, that's better. Who here uses spatial modeling or know what spatial modeling is? Good. What else am I here? Oh yeah, GIS specialists. There were quite a few from previous talks. That's great. And the last one is who is planning to use as part of their, I guess, projects. Nice. Okay. So good. That's pretty much all I had. Thank you. Right. So basically this masterclass is a little bit different. We'll try to make it a bit more interactive. It's all about species distribution modeling, or SDMs for short. We've heard of different types of SDM names and all that. But generally, I use SDM for anything to do with modeling species and their distributions. We will, in terms of the outline, we would look at some of the theory and why should you care about this in the first place. The applications of some of the different types of modeling. And then also I made some code in R, which I'm going to share, hopefully, with you. Well, I'll definitely will share it with you. I hopefully can run it here and demonstrate as we go. So we can also, some of you, whoever wants to follow, can follow that. Others can just look at this and I'll talk you through that. And then at the end of it, hopefully you will all leave with this code, the presentation, and some basic knowledge about species distribution modeling. So why should you care about SDMs? And there's quite a few points about this. It's the predictions to a larger spatial extent. So that means we're using an occurrence or other types of data sets that we collected, and we're actually predicting into the entire space of our interest or at a larger space. So also our environment are undergoing significant range shifts, species losses, and potentially also incurring huge economic and social environmental costs. So these are type of approaches that allow us to develop policies to understand what is happening with our environment at scale that is probably meaningful for policy in comparison to something small like a beach or a single environment. Also obviously helps planning with spatial planning because it's explicitly spatial by nature, so we're using GIS and various statistical or other types of modeling to create maps of what we're trying to communicate to people or stakeholders or decision-making people. And we're also using various algorithms, and these days we can use even ML and AI algorithms to model. But they're ranging from simple linear models. We're just representing those in space. So here is one of those basic approaches, is they also look in the future climate sort of thing. So we can model what's happening with the species in current time and then predict it into future timestamps and look how this will change. So we can develop policies actually accounting for those future potential changes. By the way, that's not a lecture here, so if you have any questions, feel free to jump in, ask questions, raise a hand, including people online. What is species distribution modeling? So basically, it's a process of using computer algorithms and statistics to predict the distribution of species in geographic space. So as I said, it's inherently geographic and geospatial. The environmental space is typically a climate data or other types of environmental data sets. So climate data could be indeed sea surface temperature. Environmental data could be salinity. It also could be a depth, a rugosity, or anything like that. As long as you have it in a continuous format. So primarily we work with rasters. For spatial people, I'll demonstrate later what rasters are and how to obtain some of those. But basically you need essentially continuous predicted surfaces and as environmental data to work with, and the species distribution and occurrences of different species. And then these models basically allow you for interpolating between limited number of species occurrences. So those are where you actually seen at larger scale, basically. And they could be used for conservation, the ecology, and other monitoring of impact assessment as well. I think most of it covered before. I said that we can identify the priority areas for conservation. That's, for example, areas that have more species richness than others. So there would be hotspots for one of those options. Evaluating potential for invasive. So if there is environments that are more suitable for invasive species, that will be, we should monitor more or be more prone to that. Determining potential routes of infection and disease spread. That's if you work with any type of medical type of studies. Future biodiversity changes. That's one of those we just discussed before. We can look into future provided we have those future predictions. And identify novel and emerging communities through range shifts. So that is something that we definitely experience in Australia. Lots of tropical species keep shifting their distribution. So we've done a few studies like that to kind of try and understand what would that look like in year 2030, 2050, 2100. Okay, so how to predict species distribution? Typically, it's based on the observational data. So in this case, most of my examples, I guess, from around Australia, but for the actual masterclass, I managed to get some data from India. So we'll actually work with real data from India. But generally, let's say there is observation of species from museum records, from surveys that we're conducting, from anything that basically could be citizen science for that matter. So they're usually presented as a point that has a GPS coordinates of flats and longs, latitude and longitude, something that identifies that that particular species was observed in that particular spot in the world. They, as it says, primarily point-based and summarizes also where you can get them. These days there is quite a few online databases from worms, sea worms I think that's called, to GBIF and others that you can collect, basically obtain worldwide museum data sets that have been processed relatively clean, and we'll look into some of those issues later. But generally you don't even have to leave your office to get basic species data to work with. And then once we went from those occurrence reckons, and these are the examples I mentioned for India. So my occurrences in this case are those blue points. So this is what species were actually observed. And then what I'm doing, I'm then utilizing those models that we're going to chat about this in a bit. We will basically work to predict for the rest of the environment. And typically we get two different prediction surfaces. One is something called probability of occurrence, which is those basically colored polygons or colored cells that basically demonstrate the probability that that species is going to be occurred in that unit area in particular. So it's starts from 0, which means no species will occur, to a 1, which means yes, species most likely predicted to occur in that space. And then through various methods, which I'll touch on some of them, but there is a whole body of literature about those methods as well. They call thresholding. So thresholding is thresholding this prediction into saying, well, anything beyond this threshold or below that one will be non-occurrence. That will be a 0. And anything above it will become a 1 now. So these more look like this, basically. They're two colors, right? 0s and 1s. And those 0s and 1s, the ones that we typically would use for something like creating then a polygon of where we think the algae will occur or where we think the particular habitat will happen. And that's, again, we will look through some of those workflows later. Plenty of modeling types, essentially, and this will get quite complex quite quickly. But primarily, the ideal model is a mechanistic model. So it's where the one that incorporates all the known environmental conditions, the threshold of species preferences. So for example, the tropical species that like only tropical waters versus other ones that don't like this water, and anything like that. It requires detailed data on those physiological responses of different species. If you think about the number and the biodiversity that's occurring and how many of those actually experiments been done for so many species, it's probably not many. So that means oftentimes it's just not available, these types of data sets. If you have it, work with this, it's the best available data, best data to use. Most of the time, we're just working with the correlative models, which is basically a type of statistical approaches that we're looking at the environmental sort of space and trying to separate it to environments or niches where species would probably thrive or at least will tolerate it, and the space that is not going to see any of those species. And these are the methods that most of the time use in species distribution modeling. So as well, again, data-defined models, always, all of the models basically. It's either research question, what I'm trying to answer basically, but also like what data I actually can work with, what do I have. So if you don't have environmental data, such as your depth, temperature, or anything like that, there is not many options for you except the geographical models. And it's not a single model, there's various approaches, such as the hotspots, such as the neighborhood analysis, and a few others, and we'll touch on that. But generally that means you're only using those occurrences of species, and you basically just interpolating the rest of it. So you don't really know anything else about that. You purely relying on point pattern analysis related approaches. If you have environmental data, you have much more options. So you can start from presence-only models. They call either profile models, which is basically like a bioclim model. So again, it takes, we'll touch base on that a bit more in a moment. But basically, it just looks a little bit more into the various environmental conditions. The next better one is presence background, such as Maxent, and I recommend if you've never done a species distribution modeling, start from that one in particular. It's well published. It has the original paper over 9,000 citations. So, and quite well documented literature, well documented, I guess, workflows. It's also, you can use it in R as a code, or you can just use it as a GUI. So basically you just need to push a few buttons, but as where everything else, it can get complicated quite quickly. But the main benefit of that basically is still, you only work with your presence data, and the background data that's called, so basically it's those other background points that basically extracted by the software for you. There is lots of assumptions in that, and there is also obviously lots of different caveats with that. We'll look into that in a little bit. And then the other next better case scenario is present substance. Present substance means if you have two species, for example, when you work with biodiversity survey and you survey two spots, you observe one species in spot A and the other species in spot B, that spot B could actually be the absence point for species A and vice versa. So that's an informed survey. So you basically, when you're surveying for a multitude of things. If you observe them, that's your 1, and if you don't observe them, that's your 0, basically. And that's basically within this domain, there is also the statistical models, and there's the machine learning approaches. They have both, I guess, strengths and weaknesses. We'll look into some, but as I said, body of literature on just of that is probably a couple of PhDs worth at least to understand all of it. So looking back into some of those, that summarizes also in writing those things that I mentioned before. So profile models only use occurrence data. I'll show you graphical summary of that again in a bit. Statistical models are probably the main ones that we're using, deriving different types of responses based on various predictors. Machine learning models, even more advanced things, so they can be not just linear or curvilinear, that could be basically your trees and various options. And then geographic. Geographic, you don't have species data, you don't have environmental data, consider geographic model. So environmental space, what is it exactly? Environmental space is basically environmental niche. I would use it as well interchangeably, basically postulated in the late 50s, but generally the principle of that species like certain, say, temperature, they will thrive in an optimal area, but they will be actually either absent or really suffering in those other areas. And this is the whole theory of species distribution modeling, of niche modeling, of future climate change predictions, everything like that. Again, most of the time we are affected not by a single predictor, such as temperature. We could be also sensitive to salinity, to pH, to presence of prey or predator for that matter, anything like that. So it basically gets into space, into multi-dimensional space. So in theory, your number of environmental predictors isn't limited, is only limited what you can source, what you can basically model and have for you. And so that goes from 2D to 3D cubes pretty quickly, and then could be n-dimensional basically. Another important, I guess, this, factor consider is that within the environmental niche, it's fundamental niche, which, why am I doing this? Sorry. How do I undo this? The mouse now seems to be running after me when I move. I think I just pressed one of those. All good? Okay, so fundamental niche is basically still that same niche we discussed before. So that's where species actually can occur, and that's where they most likely will thrive. So this is in this case, so that's where basically optimal conditions for temperature and rainfall meet together, and then they will be your best available space for that. And realized niche is a modified fundamental niche, which basically there is other factors, biotic or abiotic, so it could be predation, for example, that affects the distribution of species. And then even though the factors, some environmental factors are friendly for those species, due to realized niche, they're actually not going to be there for other reasons. So when we're actually predicting, it's something we should be considering, whether we're predicting the entire niche or are we actually skewing our models, I guess, and skew our management outcomes and management assessment if we're using the realized niche only. Calibrating and mapping models. So once the model is built, it can be projected. So as we say, we start with our occurrence, the blue spots, and then we project it through the statistical modeling into those different, I guess, on a raster, which is that spatial, I guess, surface that we predict. And rasters basically, we can predict it for each individual. So if we're using various rasters as a I guess, response, as a predictor variable in the first place. So if we're using the depth and temperature, for example, we can predict that either to individual one of those rasters and say the species is going to occur based on my predictions to depth. So if you use just normal type of modeling, GAMS or anything like that, that's where you hold all the other variables to average and then predict only to the variable of interest. And you can do it across all of them. Or in this case, our life, it's a bit easier. We create what's called raster stack. Or today in the morning, we saw a little bit of this cube space, right? So it's a whole environmental space that is also multidimensional because it has latitudes and longitudes. So I can predict to the entire environmental space across all of those different rasters and just have one surface of that probability of occurrence. But it actually accounts for all of my different environmental variables. And in this case, again, the green areas is basically a higher likelihood versus a bit orangey or white areas that the species most likely will not occur. And there is also, as always, caveats, small print on the bottom that there is, still may occur where even if you don't know or haven't surveyed that. And we'll talk about a little bit about survey plans, how to properly plan for those type of modeling later. Right, so I think this one is talking about bioclim, right. So this one just generally looks, that's the ones that we say this is the simplest way of creating models. It just looks into the maximum of values for variable 1, maximum values for variable 2, and kind of divides that environmental space where it says, well, because this is the best conditions for these species, we expect them to occur here, but nowhere else outside of that environmental space. So bioclim is one of those models that we can use for that. It's great because it comes with, you can source all of those predictors that are already derived for you. You can source occurrence data sets from all of those records and just work with that straight away. So it's one of those very popular algorithms as well. But as we said, oftentimes overestimates our actual predictions. And some summary for advantages and limitations, I guess. So they're simple and intuitive. They use presence only, so we definitely have that data if we're planning to model for it. Quite easy to teach as well, so convey those. I didn't use them here, so I guess that's another option. And then limitations, they are susceptible to over-predicting. So if we're trying to actually create something that is sensitive to actually good prediction, we obviously most likely will over-predict the occurrence. So if it's important for us to tell the truth, this might be not the right method for that. They cannot use categorical variables, right, just because it looks at the maximum and minimum of those values. So if it's something a categorical, it's just not ranking it properly. The benefit, another, I guess, it does not require the absence data. And then there's a couple of assumptions, is mostly developed to model species in relation of climate variables. So climate variables typically are at very coarse resolution. They could be 9 kilometers, they could be 4 kilometers. Some good models that I've seen before, they usually predicted to 5-meter resolution. So we can actually look into these differences at 5-meter scale rather than 9-kilometer scale. But again, if we're modeling ocean dynamics, then do we need a 5-meter predictions? So geographic model, those are I say that that's not based on the environmental conditions. So to summarize it, trying to use a pointer. How do I get to that bit? So changing it instead. But also when I move it, it keeps moving with me. Can I? Okay. That confuses me even more now. Can I undo it? Sorry. That's not gonna, yeah. So no need pointer, that's all right. Anyways, so geographical space model, it's one of those that takes the occurrence, no environmental variables, and we just look how those points clustered. So it's a type of clustering analysis. It can still come with statistics about your clustering patterns. You can then still derive some useful information out of this, but there is absolutely no information about the environment of this or also about the quality of those models. So we cannot validate them, we just derive patterns. And then the other ones is the environmental models, which is the rest of them. These are all the time using those environmental variables as predictors, deriving first presence, absence, or both, taking this into account and then predicting. So this is actually a prediction of the same data for from two different models, right? So if you just look into even that, this indicates the over prediction of the space that's supposed to be identified or utilized by species versus actually that something utilizes their environmental variables. And then the next one would be the SDMs with the climate data set. So climate data, we heard very great talk yesterday in the afternoon about the predictions to the ocean data, to how this is going to change, cyclones to anything really. So we do expect the climate to be changing. So it's important, I guess, to account for that as well. So typically the process works that we derive first predictions for the current climate, for the current environment, then we take those future layers as our environmental layers and predict the same model into the future. So on the left here is that future prediction of, let's say, that we predict from the current data. And then we can look actually through the geospatial approaches, we can subtract the current from the future and look how, for example, something like species richness, species diversity will change with the climate change. So we can then see some cells or some areas will have been predicted to get more abundance or more diversity, some predicted to lose it, and some predicted to not change as well. So this is extremely important for any type of management approaches and climate change mitigation approaches. A few caveats, obviously. It's still a model, so the model only as good as, I guess, the data you put through it. So yes, you can fit a model with 2 points, essentially fit a line through it, predict it on the surface, and that's your model. They're probably not great model, but that's why we need to consider what are the limitations and caveats of all of this. So because the surveys are done, museum or anywhere, there are great many studies that demonstrate that lots of times surveys occur where it's easy for us to access, where we usually go, where other people exist. So when we talk about partitioning this environmental space, if you don't have an absence data for somewhere, doesn't mean the species is not even absent there. It means maybe you just couldn't access it with your car or boat or ship. So that absence doesn't mean, the lack of absence doesn't mean actual absence, basically. So it's built for the environmental niche that is represented by occurrence record. And as we discussed, there is the actual and the potential distribution. And to best way to actually account and deal with those problems, it's appropriate sampling design. I was meant to put a couple of papers on that as well. I didn't have internet, but I can, if any of you interested to read about appropriate spatial sampling design for spatial and species distribution modeling, I'm more than happy to advise. But basically there is lots of caveats, so you theoretically need to be covering all of your available space that you're trying to predict to, if you're trying to actually comment about where species occur and where they don't occur. So it's different type of surveys altogether. And obviously the prediction should be always viewed with caution, because especially if it's in the future, we don't have any validation data. It hasn't happened yet. So we don't even know where it's going to, how is it actually going to react. So we're basically guessing. We're just trying to do the best estimate for all of this. So those are the caveats. And then summary. So understand your response type. Response type means what are you actually modeling? Is it just your occurrence? Is it your presence absence? Is it your presence pseudo absence? And there is suitable method for each one of those. And there is a multitude of them typically. So selecting the best suitable approach for your data is quite important. Use as an appropriate sampling design. If you haven't done the survey yet, if you're still only thinking, spend time thinking about how you're going to sample, how it's going to fit into your model. And don't only rely on advanced methods. So if you throw it into ML or AI, it will, even if in any type of modeling, it will give you a result. Is it good? Have you done the right way? Can you validate what you're actually predicting? It's extremely important, and I think none of the good journals will ever accept a manuscript that doesn't have some type of validation, or at least exploring any type of uncertainty around your predictions. So yeah, with using the best available data, using the sampling designs, you will give your model best chance and have much more certainty, I guess, in your modeling. Parsimony is another, I guess, terminology we haven't really looked at, but not always having many predictors is the best approach. Parsimony is the exact opposite of that. Basically, sometimes the simplest model is probably the best one. one predictor probably not enough, but also having too many, you risk overfitting your model. So you just fit this for your specific data, for your specific location that you surveyed. But lots of times, lots of metrics for accuracy for models actually provide, I guess, estimates also how your model would work in the new environment. So that's another benefit of using those models. You can use the model that you develop, well-developed model for somewhere that you fit it, and then utilize it to predict to somewhere else, basically. Use all those model diagnostics. There is plenty of them. This is just to name a few. Residuals and spatial residuals are important. So residuals, probably most of you would know from appropriate modeling. You can look at the QQ plots, how it aligns and all that. Spatial residuals, kind of the same, but then you have occasionally clustering of organisms in particular areas, and that might indicate different types of environments and importance for those species. ROC, kappa, AUC, so area under curve, receiver operator curve, all those are just methods to validate your model. Typically, it's used with the withheld data set, so we train data on 75 or 80% of our data, we train the model, and then we use the remaining data to validate it. And this is very important as well. So this is one of the best approaches in species distribution modeling. If you don't have validation, you don't really know how good is your model in the first place. Residual deviance, AIC, BIC, all of those, same thing, different modeling, different approaches. Have a read at the papers, have a look what people have done for this type of data sets, and probably if you like that paper, just use the same. There is all of them available somewhere in online packages, papers, everything like that. Another option, if you don't have withheld data, you could try those cross-validation modeling, bootstrapping, jackknifing, all of those. It just basically, instead of withholding 20%, it could be essentially withhold just one observation, feed the model for all of that, rest, predict, and repeat. So every time you withhold a different observation, and as many data points you have, you can do it for all of them. And bootstrapping is pretty similar. You just do it multiple times until you derive some sort of uncertainty estimates. Hold our data in blind validation. That's the best true test of any model. So if you get new data, you can just do exactly that. Just test your current model with the new data. But do consider the spatial autocorrelation, another term for in geography, things that are close by, more related to each other than the ones that are far away. So if basically you're collecting only points from this area and never looked there, you're most likely not doing a good job, because this area could be actually very separate, could be very different, or could be very similar. So if you're trying to predict for the entire room, you should be looking here, there, and over there as well. And it goes actually for the drone data as well. Oftentimes when we design drone surveys, it's similar type of approaches. And I guess the last one would be is get expert advice. This is literally 50 minutes, 45 minutes of background on probably 40 years of literature and research and publications. So definitely, if you have anybody that works with this routinely, methods are changing all the time, approaches are changing all the time. So reach out. Most of the people these days happy to assist, happy to provide advice, collaborate, share data. If you want to make a better model and you know of somebody that has a similar model, just reach out to those people. I think that's it from the technical perspective. I believe I had until 3, so we can probably, after this, if you have questions, happy to answer. We can then break and then come back to that actual hands-on training session. I guess I'll take questions. Yes, please.
I think that's a brilliant overview of SEMs. When I've had students kind of working this in the past, a lot of my students would fall into 2 particular problems, and I would love to get your insights on them. One is just because some spatial data are easy to find, as in online databases like IPOI, my students would chuck them all in and see what falls out. And I try to encourage my students to think about the ecology or the physiology on why that would be important. I'd love to hear your thoughts on choosing predictors. And the other is, how do you draw the line or encourage interpolation versus extrapolation. Is that an issue with SEMs? So
Yep.
predictor selection
Yep.
and interpolation or
Yeah.
extrapolation, if you don't mind.
Yeah, awesome questions. I think so. Predictor selection very much depends. So there is also correlation between predictors. We haven't touched on that as well, like with any other type of environmental data. So we have to consider that as well. Most of ML models these days can take as many predictors as you can. You can have 20 data points and 500 predictors and try to feed that. It's exactly what, I guess, your experience and mine. I usually still would limit that to something that makes sense, even though I can obtain every possible predictor in the world and check it all together and run it and rank them even, saying these were explaining. That goes to the same idea of overfitting the model. So essentially there is absolutely no reason to just go because it can handle it doesn't mean it should. And basically I would be really cautious about this. If the model, it will fit. It's basically numeric approach, right? If you provide something that it can chew through and spit out on the other side with some results, it will do it, but does it make sense? That's what the main questions to answer. And it should be answered based on your knowledge and the best available, I guess, literature and practices as well for that. And then the second one was?
Interpolation, extrapolation.
That's right. I usually bound my study areas based on where my sample is located. So if you consider something like a bounding convex hull polygon or something like that, so I don't do extrapolation. I don't like it just because, again, goes to that sampling design. If you haven't sampled there, yes, you can predict into that space. Yes, you can say that the environment will change, the climate will ruin all the species, and the biodiversity will completely disappear. That's the worst case scenario, right? But somewhere in between there is the truth as well. So if it's required, you should be extremely cautious commenting on those things. You are never sampled there. You don't really know what even to do. So for our surveys, you don't have to cover every meter of it, but interpolation is much more robust approach because it takes into account all of this environmental space that at least you sample across with an appropriate sampling design versus something that you never even looked and then you're just trying to say that it may happen. Yeah, good, thanks for the question.
Can I ask one question?
Sure.
How frequent actually you change, you give this advisory services?
Sorry, can you speak closer to the
How
microphone?
frequent you are giving these advisory services?
Which services?
The services, whatever you are giving, no? From this modeling.
Sorry, not sure.
SDM.
Yes.
How frequent you are changing your services?
How frequently you are updating your model?
I'm updating what? Sorry.
SDM model you are preparing, right? So how frequently you are updating your model, like version 1, version 2?
I think
how frequent you are doing?
Yes, I think it depends on the data. So let's think about types of data sets. If I'm working with a temperature and I'm trying to predict to year 2030 and tomorrow it's year 2026 and suddenly data is available, probably worth looking into that and updating this. If I'm working with a bathymetry, for example, which is the DEM of the seabed, how often does that change unless we go and mine it or unless we change it? So that's most likely not going to change as often, at least, unless something happened to it or cyclone went through it or anything like that. So realistically, if it's more stable environment, if you derive a good model, you probably can trust it for at least a few years. But typically, the reason to update is because somebody else went and collected more data, and not necessarily the additional environmental variables become available, but both cases apply. If you have more data, newer data, or if you have more observations, you can update the model. But the benefit of using code for all of those, it's quite easy to update the model if you already fitted the entire code, went through all the steps, then you just throw an additional data set and you just update your model.
It's not often that you see only one species in a particular area. So when you have interactions between two different species, can the presence of one species be a parameter to be used to determine the presence of the other species. I'm not talking about very closely interconnected, like symbiotically associated species. I'm talking about species like maybe like a host parasite or some other co-evolved species, or even if they are just occurring in the similar niches.
Yep. Yes, great question. I don't have an example here in my stuff, but that's in general, pretty much never there is only one species occur anywhere. It depends on what you're looking at, but let's talk about, anything typically will be co-occurring. So there are modeling approaches that you can model jointly for all of those species, and it will automatically create a predictive surface for all of the species that you looked at, provided you have some data sets. I have example on that. Alternatively, you can model diversity, so you can just calculate something like species richness or some other type of diversity index and model that. You can also, this is something, Sanita, with your data set that at least what I've seen, and again, maybe I'm not too familiar with your data, but let's say we're modeling something like benching habitat, and there is co-occurring species of sponges and corals and algae sometimes and all that. So if we have those observations, we can create something like a hierarchical clustering first, derive those clusters, allocate every one of our observations to that cluster first, and then model that again. And get assessment for each one of those classes or I guess, assessment of quality and predictions. And the more, I guess, you look across different, because some, I guess, habitats or species occur more than others, so there is also sensitivity in that, because if you want to observe all the time sand and occasionally observe some coral 3 times, so that's different, I guess, outputs and different quality of modeling will happen for that as well. Yes?
Hi.
Hello?
I just want to know a basic thing. Basically, if I'm a laboratory expert right now, we validate our results based on biochemical responses and molecular responses, right? For spatial level, how we are depending only on the physiological responses to standardize this?
Right. That's a hard question, right? It's for a few reasons. So I'm not a laboratory person, so it's harder for me to say specifically for those. I guess depends on what your environmental variables you're considering for yourself, or is it just, you can basically create, I guess, a raster for anything really. So you can, if you sample, count bacteria, you can divide your, gel into X and Y coordinates and create a raster of that, right? And then you can look into occurrence of bacteria and predict that based on something like that. So space doesn't really have to be ocean or land for that matter. With your physiological responses, I would say maybe let's have another chat about this, because I need to wrap a brain what type of environment did something like what you work with even respond to. So what would be a good predictor? Probably worth first considering, what would that be? I don't know, I don't work. What of them, give me one example of it. Yeah. 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 So I guess ocean chemistry could be one of the predictors, but depends on the scale, as we said. If you're sampling 1 point and you're working with a 9 kilometer by 9 kilometer prediction of water quality or something like that, or ecotoxicology something, then it's a problem. So you need to sample enough to have an N high enough for the right predictors. But I would say they probably exist. They're a bit harder to come by. But immediate ones I can think about would be probably, toxins, water quality, chlorophyll, something like that would be probably a good start. But also, have a read what exists. But more importantly, and this is the main conversation here, and I forgot to mention actually, if we go well with the second session, we will have some time to actually discuss your individual cases if you have questions about this and think about those things. So what would be the best predictor for you is probably based on your area of expertise and literature. How to do it spatial, I can probably advise you on that. But which ones to use for something like biochemistry, yeah, that's definitely outside of my depth. No problem.
Hi Ronan. So what would you the sample size would you prefer?
Sorry?
Sampling size.
Yeah,
So
the more the better. I'm talking about, yeah, so we'll literally, first thing we're going to look at the code, remind me to actually demonstrate the issue with the sample size, okay, and we'll discuss it,
Yeah,
so I don't want to take...
Yeah, that's what my question is, like, if it is a sample size, will affect your model or what? So, because it depends on the sample size also, your model will...
Perfect question. It's an awesome setup for the next session, so be back if you're actually interested in that.
Thank you.
It's exactly, yeah, first thing I cover is this.
Thanks.
Anybody online? There is somebody with a hand up. Right. Yeah, that's a pretty cool question. So Google Earth Engine, for those that not familiar, it's probably remote sensing. People will know what that is. Basically, Google has a server that they're uploading that raster data for all of the world. So there is satellite imagery, there is the entire collection of Landsat sits on Google Earth Engine. You don't need anymore to go on the website, download that in an individual link, select. You can literally get the collection in one line of code, the entire collection from the 70s or something like that. Then you can process the rest of it, but essentially, sorry, can you leave the question up? Because I'm obviously losing my train of thought. So yeah, so Google Earth Engine, it's a great tool. Basically, it's currently not really numerically limited, so you can actually do pretty decent stuff on the cloud without even downloading anything before you even get there. And for those of you that want to know more, that's how Josh got all of the, I guess, Global Fishing Watch data through Google Cloud Earth Engine, essentially. So that's something to consider. They put most of the fine resolution satellites there. I know the Planet Labs also have some satellite data there. So you can find a lot of good stuff on it. All you need to do is just have like a Gmail account and sign it and try that. It's probably to find, to answer what's the best important, the most important predictor, depends on the organism, depends on the environment. For benthic, the easiest one, the most important one that we always see consistently, obviously, is depth, right? So the depth itself has multiple levels of impact. So one, it's obviously the light penetration, the pressure, the anything like that. So basically, also oftentimes, at least for my approach, it's also serves as a base of deriving additional predictors. So predictors of rugosity or benthic positioning index, any of those. So those are additional predictors that you can derive from depth and get that. But again, if you're in shallow water, your sea surface temperature might be more important. But I can get depth at 5-meter resolution and using some acoustic methods and work for the entire area of my interest. You can get depth from the Earth Engine or similar services at 30-meter resolution. That's not bad, but then if you want temperature, you can get maybe a couple of kilometers of sail. So it depends on what resolution you want to work with, because the closest resolution will dictate what the resolution and quality of your actual model, of your spatial model. So that's on that. In terms of Maxent versus SDM, again, depends on the data sets. I used Maxent before. It's still a very valid method. It's extremely easy to set up and run. I'm not sure if Maxent is on Google Earth Engine, but there are other types of random forest and other types of methods there that you can fit with. Depends on the data. I would say if you have presence absence, go with something not Maxent. If you only have presence, I would definitely consider Maxent for anything. If you're just a beginner, definitely have a read and use Maxent maybe first, just to have a play, to have a look, to have understanding of a lot of intrinsics with those models. It's also very easy to follow, and it will tell you exactly how to convert whatever raster you have or whatever spatial information you have into something that's useful. Otherwise, you need to understand what you're doing oftentimes. But again, come and see experts, not necessarily me, others, but also me. Yes?
Can we use this?
Can I use these models for what? Theoretically, yes. So far we modeled up to 200 and something meters. It all depends on your number of observations and what predictors you're using. So if it's depth and rugosity and it's been mapped, then yes, absolutely. If you're trying to get something like sea surface temperature at 2000 meter, is it a good predictor? You know, probably not, just because...
Sir, as they are migrating from surface to bottom, the temperature is very
Okay, yeah, I see that one. Much harder. I haven't seen too many examples of it, but I can send you at least one paper that done it. I never done it myself. I think it's pretty hard to look into those migrations. You would need the data set that reflects that spatially, so the change over that. Theoretically possible. I can't think off the top of my head how to easily do it. Let's lump it with that other question about ecotoxicology, that I have to think about it.
Okay, sir, thank you.
No problem. Cool. Let's have some tea and come back for some hands-on. So do we have internet connection? No pressure. Now is the time. We don't. We do. We maybe do. Okay. I have code. So basically, if any of you wants to literally follow that up, most of it is on here, so I can give it to you to upload to your computers. I can email that later, or we can share it, so you don't have to do it right now. If you definitely want to annotate or anything like that, just come and see me. I can leave it here. I can talk you through it. I would like to see it back at some stage whenever you're finished, but otherwise, yeah, just let's do that. See you whenever I see you. Yep.
Okay, now we can go for a tea break, and then we will come back by 3:30 for the next session.
Till Nathan. We said let us start. Welcome back, everyone. Let us get ready for the part 2 of the modeling.
Yeah, welcome back, guys. So now we're doing a bit of a hands-on stuff. I'm happy to, yeah, as I said, these files exist, so you don't have to really follow me now directly on this, but this is basically takes us through some of the code, and I'll try to talk you through it. So just generally looking, can you guys see there in the back, or do I need to do anything about it? Is it okay? Too small, too large? Happy? Huh? Oh, yeah, okay. Well, then much easier. Right. So basically you need a few packages. For those of you that haven't seen our studio before, this is how it looks. There is different, I guess, sections of it. Generally, it's a code development environment. So this is where we develop our code. This is where we can see some outputs coming from the console. And there is a whole bunch of other windows, generally speaking. There is the plots, there's different packages that I can add, my files, and there is also the environment and other additional things. So basically, they all have some type of functionality. Again, this is not an R course specifically, so we're probably going to have to skip some of those things. But hit YouTube. There is plenty of this to start, and this code might give you also the options. So there's a few packages that I can generally recommend. I always use tidyverse for any type of simple data sort of processing. I like it. It's very intuitive and people use it a lot. We got a couple of specifically model-related packages. It's Dismo. It's one of the oldest ones and great ones. SF, Leaflet are spatial packages just to be able to create some maps, manipulate spatial data. And then SDM predictors and SSDM, that's specifically species distribution modeling packages. There's plenty of others, so it doesn't have to be these ones particularly. But I guess this is just some examples of how do you access your code, or how do you run your code, how do you access your data, what types of model can you feed, a little bit of examples of those nuances I mentioned before, and then we'll have a look. So basically, I try to create it a bit more close to home for you, for those of you in the room at least. So I've looked into a couple of papers or locations that mention, you know, local mangroves, and mention some, one of the species of mackerel, basically. So I just went on the GBIF, which is one of those museum records databases, and just downloaded that data. So you have the basic code. Typically, to get a little bit more information about anything, you could go to a help file and just basically type whatever function you're trying to find help about. And this is how typically a help file for these functions looks like. So you would have a description of what it actually does for you, different types of parameters. So this is the description up above. How would you use it in general? And then what each individual of those parameters, what the input is, and what does it serve you for. And then at the very end of this help file, you would usually have also the example of the code. So this is just in general to give some explanation. But let's see if I can answer Maya's question. All right, so here I got my mangrove data. I'll show you this spatially in a second. But I guess right now we would be looking just here on the bottom where it says dimensions of these specific data sets. So let's talk about, I guess, sample sizes and in general. So for spatial especially, it very much depends on the size of, I guess, the study area. If you're trying to study and tell us about mangroves in entire India, What do we reckon about, say, 69 samples of occurrence of mangroves? What do you guys think? Is that enough for the entire country to say where mangroves exist? Wrong. Horribly wrong. So basically, as I said, the more samples, the better. It will create better models, especially obviously the occurrence samples. So we want to know where this exists. But also, if we're going to be sampling every meter, it's probably too much, too onerous. We need to also have that balance, a spatial balance of samples. 69 is probably, again, depends also, think about any type of modeling, non-spatial and otherwise. So how many typically number of responses do you need per individual predictors? Do you remember that from your basic stats courses? Anybody? So typically a ratio of 1 to 15. So if you want to fit one environmental variable as a predictor, you would need about 15 observations to fit model that at least somehow makes sense. So in this case, I have 69, I guess, responses. So how many variables roughly how many environmental variables I can roughly use? About 4, right? Maybe 5 if I really struggle, but 4 it will be relatively okay. So if you're going beyond that, you need to think about there is all terminology that relate to this as effective degrees of freedom, is overfitting or underfitting, is everything basically related to that. So if I have more environmental predictors than my responses, I would be thinking very hard about what approach is really suitable for me. But let's say I can safely work with 3 to 4 variables and maybe 5 if I really stretch with something like that. Now let's look at the same mangrove data, but spatially. Okay, so these are all of my occurrences. So again, it's always a good idea to plot it basically in space, and I'll show you in the next sample why, particularly, but generally, so this is my GPS coordinates, this is my presence points. This tells me somebody went here, recorded this data, and put it in this specific database. So I can look and validate what essentially exists, zoom in and out, play with this sort of thing, just to get kind of an estimate and validation. The reason I ask you to consider doing this at least is because if we look at the fish data with the same approach, who sees a problem here? I'll give you a clue. It's a fish data. Hell yeah, right? So again, don't know if people see it online, but there is fish, a marine fish that is pretty much documented to be in the middle of the land mass. So if it would have been some sort of estuarine or freshwater fish, I would probably consider that. This is clearly somebody mistyped a couple of times of numbers on the GPS coordinate. So that's a problem. So that's why we always explore those data sets. We plot them spatially, we plot them, we just look at them, all of them essentially. I would be looking at, if you remember, the dimensionality was 118 columns of something, of some type of data. Do we need it? Do we not need it? We need to decide about that. Essentially, what do we keep, what we don't keep, and this is not part of this course, basically. But something like this, I cannot proceed with a spatial model having fish represented. Because think about, I'm telling the model that my occurrence of fish predicted to be here, in the middle of the continent. So it's a big problem, and I didn't do it. This is directly from GBIF. So this is directly from one of the major downloads from, and I didn't even look for that sample. That was literally the second data set I ever accessed. So always, always, always plot, try to question any of those data sets. So what's the ways of deleting that or removing this or keeping this if you think it makes sense? But how would you, what would you do with this essentially? How would you deal with this? I would remove it, right? So there is a couple of ways. So I would say the hardest way probably is actually trying to identify which record that is, how to ID it, access that, and then in R obviously you have a simple example of just removing by row, removing by name, removing by some sort of unique identifier. You can remove by, I would say, something like longitude. So if you say if it's between this longitude and this longitude, you can probably clear that. Another example. I'm too lazy, so I'm not going to be doing that. But the other option essentially would be an easy one as well. I'm working with what type of environmental data? That's right. I'm working with marine data. So if I'm deriving now, I'm making a database now or data frame that will have my occurrence record, their individual GPS coordinates, and then I'm extracting a bathymetric or let's say SST, sea surface temperature value for this point. What would that be, that value? Wouldn't. NA, right? So it will be basically NA, so it doesn't exist. So that's the easiest way to remove it. So I just go, I will show you later how the rasters look and how this data looks overlaid over some sort of environmental data. But generally if I don't have a predictor value for this, it's not going to be 0 because theoretically you can have temperature of 0 if you're in the Arctics or anything else. So it would be NA. And then in the modeling, one of the simplest one is just NA omit, NA remove, NA anything else. It just excludes this automatically. So, but not every modeling approach does it. So just keep it in mind. So you still have to probably rather identify and remove it. This time I'm lucky, so I'm not going to be worrying about that. Layers. So same thing. So layers. BioOracle, that's another sort of global database. So I'm accessing this through those packages that I demonstrated before. I think it's one of those SDM predictor packages. So it just basically gives you a description and a whole bunch of different layers that you can basically use and look and understand. It also has some of the future layers, but I'll talk you through that in a little bit. But let's say just so for the sake of this presentation, I'm accessing the SST, which is the sea surface temperature one, and I'm accessing another one later. But basically, this section of code is, without going too much into depth, it just basically access this BioOracle database in general, look into what names of the layers I have. I extract the specific SST layer as a mean, I guess, based on this SST mean. So I'll load this into my memory of my thing. And the last thing I do also, I don't need it for the entire world because this is what you access essentially. This is where you're getting. I'm just cropping it to the extent of my study area, which is roughly this. So just to area of my India, I guess, continent and the ocean around this. And then I'm just plotting that, and I'm plotting my points on top of it. So again, just to validate, what does that look like? Yes?
Temperature data from another. So how do you correlate? Because that is a different time.
Yeah.
And this temperature data, do you take it from?
Yes.
How do you do that?
There's few options. So I'm just, again, being lazy on this one. So I'm just going by the mean. So you can get this, and depends on the resolution of the data sets. You can get the manual, seasonal, monthly. Okay. Depends what you want to do. It's absolutely great question. Actually, I should mention that, obviously. If you're modeling from data of species from 2020, I would be looking for the SST from 2020 specifically.
So when I take the data of the biology, I take it from a particular year, not whatever the presence data is available in the database. How do you do that? Are you taking suppose this mangrove data, species data. So I only say that I want the data from 2020, whatever presence data is occurrence. You don't take the entire database, right?
Well, there is only 69 occurrences, so there is not many choices we would have.
Yeah, okay.
But I think in your case, if you're running surveys for 17 years, you probably have much more data. So yeah, so I would be, you could subset, there is thousands and thousands of those layers there as an example. So you would probably can subset by year basically and extract, yeah. You would just need to create a database of SST based, so you would have, I guess, year of your survey matched with the year of the SST. And then again, you can get, in terms of the SST, there is the mean across everything. There is max and means for different years, and I think it goes from 2000 or something like that. I provided all the links to all of the data sets in the bottom of this file, and there is online, there is basically a description of all of this as well. So I would definitely need to hit, I guess, the help file on those individual data sets to work out what what's there. I might be able to do it here. Let me see. See if I can do it in real life. What screen do you think people can see now?
Maybe I'm going to interrupt while Ronan's working on that and suggest that I think when selecting predictors or years of predictors like SST, really thinking about the physiological or ecological reasons on what might drive the presence of those species. I work a lot with fish and invertebrates, and where a particular sea star might occur might be more determined by the last 2 years of temperature than the last 30 years of temperature. So I would restrict my thermal properties to what I think is most relevant to the physiology of that occurrence. On the other hand, if we're looking at mangroves, maybe where a mangrove occurs might be due to the last, 50 years of temperature, or maybe seasonal averages. So I think having a really careful think about the ecology and physiology of a presence, and then selecting the time frames of the predictors that best match up is hard.
Absolutely, yes. Especially with those, seasonal data sets, they definitely should be matched. So if I'm completely desperate, yes, I would go for a whole mean of everything. But then if you're trying to, I guess, create some sort of temporal variability predictions, you would have to match it to the temporal sampling of your data sets. But this is just a quick one about the BioOracle data overall. So there is ways of downloading this manually as well, and in the help files there's also how to integrate it with R and Python, which I would rather recommend. But as you can see, so there is ocean temperature, there is any types of options for that. I'm not sure, I don't think they have like annual actually, they just have, maybe by every year, and a whole bunch of other ones. So basically you've got different types of chemical parameters, pH, chlorophyll, but generally this is one option. And then up here you also have a little bit of future predictions. So again, depends on granularity, and the pH depends on on what you're interested in. So basically by decade give you sort of average predictions of that. These ones are a little bit different to load up. So the previous ones I can programmatically access through R and just stream it directly and download and load this into mine. These ones used to be, but no longer at the moment. I suspect something to do with the US turmoil that they're going through. So you would have to download them basically individually. And those are netcdf files. So I don't know how many of you worked with netcdf files, but that's, yeah, so they kind of a bit tricky to work with, but generally they would be quite same. They would have quite a lot of information, spatial and layers and all that, but more than happy, to advise on that at some stage. For this practical, I basically already done the download and excluded that. sorry, I already upload this in the memory of my software, so it's basically sitting there. But generally, the steps I went to BioOracle, clicked on the decades. I think I downloaded a 2100 sort of thing to just show the stark differences between predicted distributions. Download this to my computer as netcdf, unpacked, extracted, and plugged it later into this one. Okay, so going back to this. So just to recap, so I have my occurrences of mangroves. This is my mangroves here. I have my macro occurrences just here as well. I accessed and downloaded 2 layers, my SST and my chlorophyll, both means, but that's just for the sake of this essentially exercise to not overcomplicate things, which could get complicated. And then last, I guess, operation I need to do before I sample and feed the models is essentially I create raster stack. Raster stack comes from the raster package. Basically, it's all it does is basically if you visualize, so basically it takes SST, then layers this over my chlorophyll layer. So then I can sample them at the same time with my point data set. So it's just easier to extract. I can go and extract those point data for each individual predictor or environmental predictor, but to do it easier and quicker and also work with the predictions from the model, I create those stacks, and that's what I'm using. So basically, this is my current stack. I have those 2 layers in it, and I plotted that, and I plotted on top my data points. So again, I work with macro just to keep it. So that's the demonstration that I wanted to show you. So basically, it's still there, but then when I extract it, actually, it's not going to get any SST or chlorophyll data because I physically don't have it there. If I had it, then you're in trouble. So pay attention to what data set you put through that. Okay, in terms of modeling, so there is a couple of, again, this is all comes from package help. So I basically I got that package of SSDM, which is a mixed ensemble modeling for species distributions. So there's different ways of doing this. I can download something like a linear model package and fit it through that. I can download a random forest, for example, model, and which is a machine learning one, and fit it through that. I can do a GAM individually through different types of packages, GAM4, GAM, GAM-MM, anything GLMM, anything like that. But this, one of the options essentially is doing an ensemble modeling. Ensemble modeling, the benefit of this, it's you can fit different models. So in this case, I will be fitting 3 of them all together for currently just my species, which will be the mackerel still. I'm working with those ones. And then within, I'll talk you through the code a little bit. Okay, so first of all, I need to create from my mackerel stuff, I need to convert my data frame into a spatial data frame and create the right columns to fit into those models. Doesn't matter right now, but all you would need to read file on this ensemble modeling function to understand what you need, and that will tell you what type of data do you need. So I started fitting that. It gives me an error, for example, like, oh, you're missing something. Then I need to go and troubleshoot it, and I done it by basically converting my initial data frame into spatial data frame. Spatial data frame, and then that's what becomes essentially my input here. So in terms of ensemble modeling, we have algorithms, and then you can choose. I think there is at least 7 or 8 of them. It also, again, comes through the help files. We can look through those. sometimes you have to scroll quite substantially down the help file. You can also open these types of PDFs just in a different window altogether. But here we've got 1, 2, 3, 4, 5, 6, 7, 8 or 9 basically, different algorithms. So as we say, different one of them have different caveats and have different assumptions. So be careful with this type of modeling. I'm not a big fan, truthfully, of like just checking all the models, all the data. But this is, people do it. There is lots of published materials about that as well. But, you know, as we discussed, these ones assume that you don't have any presence, absence data, so it does kind of pseudo-absences. This is a purely linear algorithm, so then if you have data that a little bit more complex kind of doesn't really work for your data set, this is pretty good one. Then this one, similar assumptions, different approaches. Each one of them has something, so basically you have to understand what you're using. So in this case, I'm just fitting 3 different ones so we can talk about them. I'm just doing this in multiple replications, replicates times 3. And I'm doing my cross-validation, which is kind of, as I say, it is quite important. I only have 69 records, so I wouldn't be able to apply the usual split of 80% to feed the model, 20% to test it, or 70-30, anything like that. But this one basically says every single time I just I save one sample outside of the 69, fit with everything else, and validate with that single number. And then just repeat it, and repeat that 3 times as well. So basically I validate my model, whatever, the 68 times 3, whatever, times in third sort of layer. And then that's that. This is occurrences. This is my data set. So it basically tells the model the lats and longs of my latitudes and longitudes of my occurrences. And then because I only model one species, so I don't need to describe and tell them there is multiple species in it. Thresholding, we discussed a little bit. That's the way how to convert your probabilities of occurrences into presence absence data. So lots of literature, important thing to consider. I only have randomly selected 0.75, but it could be vary from 0.2 to anything really. But that's what tells you where the absences stop, and I guess presences do it. And this is similar to that one. So thresholding approaches, there is different reasons to apply different thresholds to it. So I can apply it because I want to be more conservative, less conservative. I want to, if I'm making a marine park, I want to be maybe less conservative because it then over predicts distributions. If I want to be more conservative, I can do that as well, and then hopefully that will be a bit more helpful. But basically, all of this exists in this help file with links, and further down the line that you can dig into all of those essentially different methods and try to understand that. But realistically requires lots of thinking, lots of understanding on the back, which is when talk to your local professional comes too. So then basically to fit it, I just need to run that section of the code, which I'm pretty sure I already did. So I just go straight to the model results. So fitting 69 samples with 2 predictors doesn't take too long, a few seconds really to fit it all. And then, oh, you guys can't see that. Okay, we'll do this and that. Right. So these are roughly the same sort of maps that I had in my talks. These are literally, I've done a script grab before, but basically these are outputs of those models. So there's quite a few of them, and again, reading the paper which I have in the comments down below about this entire package will help you also understand and decipher these all points. But as we said, so first of all, it's that habitat suitability, which basically defines my probabilities of occurrence of those different different species. So I can see wherever it's greener, it means it probably gonna have a higher chance of occurrence. On this side, we're looking at variable importance. So I only use two, so chlorophyll and temperature. So it shows me that my chlorophyll predictor are actually quite more important, significantly more important, I guess, than my other. Obviously, if I had more of those, it's probably going to be more of those. In this case, because it's a macro, so at least from what I read about the local macro, they actually like quite silty mangrove sort of near mangrove area and with higher sort of proportion of silt in it. So it kind of makes sense to me because at the end of the day, we're still looking at the tropical species, but the story could be different if you fit with more samples or anything like that. So these are the two main ones. Then going across the tabs first is the binary map. So that's that conversion from thresholding, which gives you then 0, 1 sort of predictions on that. Uncertainty, it's one of those same thing. Because I've fitted 3 times and on 68 samples every single time, so basically it creates uncertainty map of those, variability, and every single one of those, it's actually a cell, right? So it's a cell of a raster. So basically it predicts every time and looks if it's consistent or not, calculates the difference between those predictions, so sort of like the residual, and then just plots that essentially in space. So if I have higher uncertainty, means my prediction is possibly not as good, so I cannot trust it completely. I still have to worry about this. I think overall this is still a good numbers. If I start heading towards 0.5 or something like that, that means we're basically working with 50-50 chance. So I'm just might as well guess where my species will occur. Summary is some of those model parameters essentially. Here on the other side is roughly same. It goes through the variable importance now. So we have the bar plot initially. Now we have this in numerical summary, and the legend will give you, I guess, what is it all about. Then on the bottom panels, we are looking at variable correlation. So we can see between the models, right? So we see that GAM and random forest are quite correlated models. So is there a way or point of using both of them? They're almost 100% correlated to each other. So maybe don't even need it. Maybe you want the agreement between models to be higher rather than lower, because otherwise, if your models don't agree, then basically it creates more uncertainty. So perhaps you would want to keep that one. GAN and GLM obviously quite similar as well, but then random forest and GLM have different predictions just purely because they're different algorithms. One RF random forest is quite sensitive and can adapt to different data sets. It's a machine learning algorithm. The other one, it's a linear model basically, so clearly different variations. And then model evaluation. So that's some of the methods we mentioned before, the AUC and the percent cap model. It's all sorts of different types of model validation algorithms and approaches. You will have to read. It's quite intrinsic and hard to explain in one go, but basically AUC, typically a classic comparison or model validation algorithm. AUC and ROC are the most important, most common algorithms to validate any type of presence-absent data or presence only. Kappa is usually, if you use something like kappas, are typically for classification models. So it looks if you're doing like a land cover classification. So it looks if your predicted values align with your observed values. And then if you have continuous data like species richness or something like that, then you can just look at pure correlation between predicted and response values. And then maybe do like a R square on it or something like that, or different types of that. So that's kind of the main components of that validation. So this is quite important, super informative in that regard. And then going back to the code, I'll just show you quickly that basically I can also access some of those. somebody has a question, so we'll let them ask in a second. Yep, we can ask now. Do you want to facilitate? There is a question online. Right, maybe let's keep that. That's not directly right now relates to this. I will answer that, just if you guys can remind me. Yeah, sure. I would 100% would not use the model, first of all, at all. But yes, how to improve the models. I think we had some conversation here over the break, but more data, definitely. Changing predictors, absolutely, because clearly your predictors don't work for your model. I would also though consider, because it's a spatial model, so you would need to also consider resolution, the spatial autocorrelation, the stuff we discussed before. So the resolution, the implication of this, if your points are close by to each other, they most likely will be falling within same cell of that raster. So you're actually having the same value of your predictor for all of your observations, for example. And that's obviously will create a bad model. So that's why some of those processes are very important. I'll just go back. One of those things doesn't work. But I'm not too stressed about it. Am I sharing? Yeah, I'm sharing the same. Okay, so one of those just doesn't work at this stage, but that's okay. Basically, that was just showing before. This one. So ESDM, it's my model that I fit, it's the object. And within that, there is different attributes that I can access individually. So in this case, I access evaluation, which is some of the bar graphs that we've seen. So if I want to put it for my report, instead of doing screen dump or grab of that previously shiny sort of application, I can just put that, output this to my screen and just have that as a, or use it somewhere later in the report. So that's what it does, basically. Okay, so we fitted a model that ESDM for the current climate. Now let's predict for the future climate. So as I said, the future predictors, layers, come from the BioOracle as well, but I couldn't access them through code. So basically I went and downloaded that, uploaded here, and now I have them in my object. And then in terms of those, I've created the same. So if I create this stack object, future stack in this case, so previous one current stack, this one is future stack, and this has my layers named exactly like the previous ones in my model. So otherwise it's just not going to work. It has to be the same predictors. It usually also has to be either of the same extent or same resolution. Sometimes it would take maybe different resolution, but not the same extent. Sometimes it's something else, but it always will give you some sort of error, which you need to resolve. I had to, for this, resolve that extent error by just basically creating the same extent for all of it. But otherwise, so okay, to disappear that for me. Oh no. Right. So yes, basically this would work with most of the spatial data. So what you mentioned here is probably JS formats. It's rather than formats, it's actually you mentioned different JS software. So QGIS is a JS software. Shapefile is basically, again, comes from Esri, but it's not even a raster. So short answer is probably not that. But essentially any type of spatial data, if it's in a raster format, which is like your geotiffs, your ECAWs, all sorts of, so basically it's same like Umberto was showing, Dr. Umberto earlier today. So it's basically like gridded type of approach, and each cell should have a value basically. So that's what a grid, raster, a DEM or something like that means. But yes, absolutely. So if I get a model predictor in a GOT file, which is quite often what you get from lots of those places, I could transfer it to ASCII file, ASCII, which is what, it's just like a text file that's basically required by Maxent, for example. So there is geospatial packages in R. You can basically go in Esri and convert that to something. Most of QGIS and Esri are quite interchangeable, but some of them are a little bit harder to format. So basically I have both of the applications on my computer, and sometimes if one doesn't work, I just have to the other one and then tweak it basically. But yes, you can generate between different ones. Sometimes a particular algorithm asks for a particular type of file. So Maxent only would work with ASCII files. So if you don't have it in that, you have it in GOT, if you would just not take it, as an example. Okay, cool. And then back to this one. So we've got our future stack named the same way as the previous stack. So that's what will facilitate the future prediction. But when you have a chance, go back and look at the predictions of temperature and chlorophyll from the future and from the past or current, I guess, stack. And you would see that they actually quite vary. So the actual values everywhere will be different, and that's kind of the point, right? So we're looking into the future predictions. So I have that. Then next one, I'm using basically that stack. I'm converting this basically. Again, that's what I was saying before, that I had to create same resolution sort of thing because it was giving me an issue. And I kept it for you just in case you ever run into those problems, so you would know what to do. And then back to that. Okay, so this is my future predictions. And again, just for comparison at some stage, go and have a look into future and current predictions. So basically, obviously my observation points are the same observation points, so I don't have anything else. But my raster predictions are vary, and it's based on the new values that I receive from the future predictions for SST and future predictions of chlorophyll. And then I can, again, go through the binary maps and through the importance, right? So before we haven't seen that yellow, right? If you start zooming on these areas, you will see that it's different. But at the end of the day, again, I think this is now we're predicting for, pretty sure it's the mangrove. So obviously not expected to be seen in the ocean, which why you get nothing realistically. Okay, and then same thing with model validations. We already seen of this. So right now, model correlation is quite low, which could be good or bad, depends on your view of this. And then the AOCs and everything like that else, kind of good, but I would be digging deeper into all of this, basically. But again, we don't really have validation for any of this because we're just using this purely for prediction. So we're looking at the year 2100, so we don't have data to validate it. It's basically a guess. Back to the code. Another options for predicting stuff is the stuff we discussed before, multiple species, which is, of course, how could we not consider that? So now I'm basically bringing both. I already have my data from the macro, and now I'm joining the macro with my mangrove, basically. So yes, it's not going to be everywhere the same, but some areas will have now overlapping sort of distribution predictions. Those intertidal ones where the macro will still most likely will occur with the mangroves. So we still can do it in general. So then I have a new data frame, and then I predict that. I'm, again, using the current stack, so I'm predicting looking into 2 days. I'm using my similar models as we looked before. I'm looking now at multiple species. Still, longitude and latitude, that's the basic thing I need for all of it always. And then my species column is the name. So one time it will be a mackerel, one time it will be mangrove. So that will combine them. And methods in this case, again, there is different types of methods fitting. Let's not look into that one. But basically, there is different types of methods. So it's basically how you weigh those co-occurrences, because sometimes it's going to be this one, sometimes it's going to be that one, sometimes it's going to be both. So this is what it does, some of these methods. I asked this time for some output to the console, so we can also have a look into that one. And I'm using the same threshold for the binary reasoning. But again, reasoning could be changed now because it's now multiple species. Maybe I want something else out of that output. I fit my model, and I'm just now going to plot it and look at it. Sorry, I realize I have too many screens now. It's still the same. So one of the outputs I am interested is that species diversity, is essentially, so where are the locations, and this is just for 2 species, but let's say where are the hotspots, basically. So this is showing me the diversity index, and basically it just categorizes every one of those cells that we use as part of the prediction to tell me it's going to be either nothing, a 1, or a 2, because we only use 2. If we use 100 species, you in theory can go up to 100. And look through any of that. And then this is your biodiversity essentially estimate. Pure, I think it's the alpha diversity sort of assessment. Again, comes back with some of these, the plots, the variance and contribution of different types of predictors. Again, I only use 2 at this stage, even though in theory I have now twice as many observations. And then on the bottom, a little bit different story again, because I applied for a different, I asked for a different type of model selection, model validation. So we're looking into our kappas, our specificity and sensitivity, which is quite important as well, which one tells you how good is your prediction of presences, and the other one how good is your prediction of absences. And we obviously see that there is some variation in all of that. And again, the rest of them requires a bit more of a read to understand what the intrinsics are. You don't always have to use all of them, but let's say again, specificity and sensitivity and kappa is kind of common, very common. So I'll be definitely considering something like that. The other ones today been mentioned, F1, it's another one to validate your model, which is very similar to sensitivity and specificity. It just takes into account both as one. Kappa is kind of the same, but just different type of calculating that. And your model, I guess, overall, I guess whether they converge sort of thing or not. So the output pretty much the same, but the point of this, it will be now the diversity of predictions, essentially. So how many of those you get. And I think I have more of this. Right, so that's the stuff that didn't work before. Similar stuff is here. I can independently, through the initial object that plots in a separate app or something like that, I can access these individual maps, metrics, everything like that. So I can just save this map or save this layer and then plot it in my Esri software, in my QGIS software, anything like that. Or just keep it here to the console, basically. But it's the same map we saw before with the predictions of 2 species. It's shown here. And a couple more, I guess, options from the code would be this time is plotting. So basically it's the same. It just summarizes all of those predictions, success and kappas and specificity and all that. So I can basically create tables, put it into my report straight away, or save it essentially for a CSV or something like that for future validations. And then the last one would be, again, NDMIS map. So basically another metric that can show you which ones are sort of areas for specifically species just occurring there primarily and just there only. So it goes to endemic species. I think that's primarily all I had. There is some resources, so MaxEnd paper with references to all of the software, the GUI, the information, everything. I still very highly recommend that for using that, just because, again, people use it all over the world, nothing wrong with it, just have to be really conservative and understand what you're doing in that regard. BioClean package is another one, is the one we discussed before, that you don't need too many things to fit and easy to teach as well. Environmental data sources, there is a couple. BioOracle MassSpec is primarily for something like bathymetric data for the whole world, basically, as well. And then there is WorldClim, which is if you work with terrestrial data, I think a couple of people here are, or by association. So it's like BioClean, but just for terrestrial data sets. The paper that this whole thing based on, with some additional explanations of things that I didn't cover. And I still owe you some survey design papers. If you're interested, just hit me on the email, which is somewhere. I can give it to you later. Yeah, I think that's pretty much it from me. So now we can do some questions. We can, if you want, we can chat about your independent individual data sets, or we can just wrap it up and go home. Pardon? Yeah, sure. Of course. That's right. I was trying to avoid it. My objective is to predict future distribution, habitat, suitability of species X by, right, what's the historic period range can be considered for the environmental variable. So the first bit, what historic period range. So basically I would model, as we just saw, the current distribution based on what I have now. And then your 2045 depends, because I doubt you will find individual year predictions. You most likely will find something by decade. So you could look maybe 2030, 40, and so on. But I would, same thing. So BioOracle is one of those I showed you today. I'm sure there's more. The WorldClim would have future predictions for terrestrial data sets. And the more important thing, and I think most interesting thing, is maybe we could have a discussion about what do you have available in your country primarily, just because in Australia we have additional resources. So some of you know of CSIRO. So we have some of their prediction models. So it doesn't have to come from any of those websites. You can, in theory, take any type of future prediction as long as it's in a raster format or created, converted into a raster format, and use that in your model fitting and model predictions. So maybe once I finish answering this question, the second part of it, we can chat about, where else would you go to find additional data sets, additional layers, for both, for occurrence, but also for environmental predictors. And also how this DM model validation can be done without the field visit. Right, so touched on that a little bit as well. So to fit it, somebody had to be in the field at some stage in their life, right? So you have to have occurrence data. So it doesn't have to be you, it doesn't have to be anybody from your organization, but you need to have an occurrence data, otherwise we're not fitting species distribution modeling. So that's the way to do it. So typically, if you don't have it, somebody would. If you can't find it online, try to understand which organizations in your country, in your environment, work with this type of data. Find the right people, contact them. Today we had a short discussion about small n for fitting anything like that. So my advice was, well, who else would have your data set? So contact those people, collaborate, you know, and pitch what are you trying to do, and people could, most of the people these days pretty much happy to share their data, especially if they promise some sort of output, not just, you know, a black hole of nothingness. So yeah, so that's how you go without having any of your data. So historically I used other people's data. I often do it still, especially if I'm looking through the, you know, country sort of averaging or regional sort of modeling. I don't have the time or the capacity to go entire half of Australia and look into that. I just use the existing data sets from other people. And as I said, there is global ones, but there is most likely in-country ones. So who knows about anything in the country here that you would go to look for your species occurrence data sets for anything marine? Anybody?
Is it possible to get a temporal trend of the prediction?
So across years, you mean, sort of?
Yeah.
Yes, so you can get it as a mean, I guess, overall. If you, let's say, species richness, so we can look at the decade 2040, decade 2100, and decade of now, or something like that. If you predict, again, if you fit temporal models of essentially, say, seasonal or something like that, so same thing. You can summarize, I guess, either per area, so, you know, the entire sort of coastline prediction, so you can summarize for entire. You can summarize for areas, essentially, so you kind of, that's a geospatial processing, so you're just looking at the predictions within specific area and just average that, or you calculate mean, range, max, anything like that, and then you just plot it as a time series thing. I would recommend doing this still through mapping, so you can basically create predictions for each one of those throughout the years and just do as a multifaceted graph to show the difference from year 0 to year 1, year 10, and so on. And those are, yes, some great papers about that.
And thanks for that. And usually a lot of data that we get about historical species presence in a different place is through secondary data, or it's from like questionnaire surveys. So there are these people telling us there are these species here in this place, in this region,
yes.
and it's not there now.
Yeah.
So, you know, this is still data, but how can you pinpoint a particular location, or can you give it on a spatial scale, on a 2D?
Yes, I think we're talking more in Australia, we call it traditional knowledge. And it's hard, it's a little different approach. So the way to deal with this for us was go with a big map, essentially, to people and have community engagement where people draw what they think, a polygon on a map, saying here they occur, here they don't, here is a rock, here is something else. There is a lot of, I guess, intrinsics involved in that, because some indigenous people in Australia call corals a rock and they don't differentiate and don't care, but they care about the fish, so they would know exactly what size of the fish lives under that rock. So it depends what, but the good point is basically doing community engagement. You get 10 people, 10 different experiences, but at some stage they actually start overlapping. So you would actually can create those maps sort of where people do believe of a hotspot of something. And then we've done some of the previously validation from those maps, and they actually quite well correlate to some of those models. So it's completely possible to do. You need probably a geospatial person on a team that can envision how to create it into maps. I find maps are very easy to communicate, right? So even if it's drawn, or when I go in a field, I oftentimes just I have a, I don't know, satellite sort of imagery of the area, and then I just, same thing, draw on a map what I see. And it's not precise, clearly, oftentimes, but it's better than not knowing what they're in the first place. Sure.
Sir, what is your point of view during the model preparation regarding extreme events like tsunami and cyclones?
Interesting, yes. They hard to capture, that's my point of view. So they typically, you would need probably to convert this spatial data into something more, I guess, of a graphical representation and overlay it with your event, a peak of your heating event or something like that. Unless they capture this in a spatial sense, in my opinion. So you would need, somebody basically capturing those heat wave events, for example, in your spatial data sets, which typically, if we average for the entire year, I can guarantee you just get smoothed out. So it's really hard to pinpoint, but so is like, I guess, storm events on the beaches and beach erosion related stuff. When Marina is looking at all the debris, we're actually trying to tease it apart right now because we're looking at the general beach profile, which changes seasonally, but also every type of storm or cyclone event either dislodges a lot of plastic or dumps a lot of plastic on it. So same thing, we're trying to look at the spatially at the entire thing, but then this extra type of cyclone or storm impacts could be a different type of approach and consideration. But especially it's really hard to account for it. It's pretty much impossible as far as I know.
Yeah, thank you for presentation. You are covering the habitat susceptibility map and uncertainty map. What is the resolution of this model SST? Means SDM model?
Yep. The prediction of this model is exactly the same as the resolution of the initial layers. So that's whatever comes from BioOracle. I cannot remember, but I would guess it will be either 4 kilometers or 9 kilometers. Cell, yes. And that's, again, comes down, if you have a different model, a better model, an elevation of 30 meter, then you can predict to that. The point is, though, if you have a predictor of 30 meter, an elevation, and a predictor of 9 kilometers, the better approach is actually, essentially, you can either subsample one to downscale 9 kilometers to 30 meters, but then you will get a lot of cells of 30 of the same value, or you can, I guess, upscale the other one to create coarser resolution from your, but they all have to be the same resolution to work. And typically the better approach is essentially, it's actually coarsen the resolution of the final layer. So your model resolution and predictions are as good as your worst layer in your data set.
Thank you.
Yes?
I have one question is regarding when we consider biological functions like growth, lifespan, and this complexity will change over the time, maybe in a season or maybe in a year. Say for example, we are going to give a prediction for next 10 years. So we may be crossing several generations of these marine organisms. So how these lifespan and other biological functions are taken into account when we predict the future for next 5 years or 10 years?
Yes, that's an extremely hard question. I have modeled length, essentially. So there is obviously the fisheries approaches to predict growth based on temperatures. They're just non-spatial, but you can still create something like that. To capture this spatially, one option would be, so you can model length of individual, say, if you look in fish, you can model length observations from your observational data, make it as a prediction. Then let's say you expect that this will increase by or decrease by X amount, 30%, 40%. Then you can basically, the easiest one will be creating a second layer that you multiply your original length layer by a nominal amount of, let's say, 30% increase in the size of a fish because of the warming or something like that. So then you create a next prediction layer that is fish predictions 30% higher. but it doesn't have that variability of fine spatial resolution of where you actually can say, yeah, this is actually going to happen. It's within the same range of predictions of the future scenarios. So basically, it's a guess. Nobody knows if the actual temperature in 2100 will be what this currently saying. We don't know that. And it's the same goes for any type of these layers. You just probably have to find either an average or regional predictions what they will be, divide your map into those regions and multiply each region through the spatial operations by a certain amount. That's how I would approach. I might be wrong, there might be better ways to do it.
Yeah, you are shown that SST data, is it in-situ measured data, means that bio-optical data, is it, what is the accuracy and what is the resolution of that?
SST. Yes, SST. So again, those are not data sets that I derived. They're coming from the global, I guess, predictions. I presume they all would have that. So SST data these days come from satellites, so they're pretty good and well validated, but I'm sure they vary regionally, essentially. So they might be performing, most of those global models perform well in the open ocean. They oftentimes fail in areas where coastal. So especially something like prediction of temperature, even current temperature sort of thing. Any of the metrics of water clarity or anything like that. So chlorophyll, all of them, big challenge for all of the oceanographers and remote sensors in the world is predicting those in coastal, because coastal process is so complex that it changes hourly, daily, you know, and even if they produce a few hours of that, that's kind of hindcasted. So there is a lot of variability in that. But if you're doing something of a larger scale, you can trust it, but it comes down also to best available data. So I would always look what is there, what is available. I wouldn't trust directly, you know, BioOracle or BioClim or any of those. I would look what's more available. I would first start looking from local and regional data sets, and if I don't find those, I would go to global ones.
Any particular situation to reduce this accuracy of this model, SDM model? Any specific situations?
To reduce the accuracy. Yes, absolutely. So the easiest one would be, I guess, again, if all of your data comes from one cell of that model, if you sampled only, you know, in your raster sort of in one area and all the predictors are very closely clumped, then you basically don't have the entire environmental space covered for this. So that's kind of goes to poor performance of any model, which is where all the sampling design and thought process about how to sample your space is very important. Using, again, data sets that are misaligned, I guess, in terms of resolution as well. So, you know, if you basically sample or upsample one of those, then you're changing essentially the resolution of the originally intended data sets because they tuned for a specific use. But it shouldn't stop you to use it. You just need to acknowledge and understand the limitations of those models. I would still use it because otherwise we cannot predict anything really. But not anything needs to be spatial, and some of those just needs to be worked out and improved.
Generally, models are applying for a particular reason, and globally, there is the accuracy depends on some of the parameters. In this model, what we have to consider for applying to Indian region, majorly?
Yeah. So definitely would look into any of the layers, like what is the accuracy, but I guess nominally I accept that they're good enough to trust because the scientific community uses them. So if you source this model from somewhere else, like a SST or something from something else, I would check obviously where it comes from, but there is quite a few of them that are globally acceptable. So that in terms of reporting, as I showed before, you can validate your actual species distribution model, but primarily the sources and uncertainty in those models come from species data more so than from the environmental data. But to know how to wrangle that environmental data is important, and that comes to that resolution extent source of those things in the first place.
Thank you.
Thanks. Sure. Sure. Luckily, I've got a larval ecologist here. Maybe he can address this question.
Yeah, that's a really good question. Then I think there's two parts to the larval nursery habitat question challenge, and that is larval availability and larval delivery, which is hard and dynamic. But for the habitat quality itself, absolutely, particularly if you can have some good sample data like Ronan mentioned, and empirical data on where the larvae are recruiting to, it can absolutely be used. I think the physiological and the environmental parameters to drive that will be hard, but the approach, the framework, the statistics, absolutely applicable.
I will just add one thing in consideration, is depends how you also collect your larval data, because there are only two ways I can think about. One, it's netting, and sometimes you tow for quite a prolonged amount of time. So if you're trying to relate it to a fine scale habitat, and you tow for 2 kilometers, so you typically will be averaging for those 2 kilometers, because you can't tell that if this larva came from here or 2 kilometers upstream. Similarly, if you're using the other way of collecting larvae, it would be light traps. So larvae has a quite substantial swimming ability, especially in the more developed stages. So they might be attracted to that light from a couple of hundreds of meters at least, as I can think about. So again, if you're using a 5 meter some resolution of a predictor saying this came from here, it's not necessarily true. It could be coming from kilometers away. But otherwise, whatever you said.
Dr. Ronan, I have one question.
Sure, I can't see.
Yeah. It was a great presentation, by the way.
Thanks.
And like you did say about there hasn't been any validation because we don't have future data in our hands. And I think in the last presentation of the morning, we had modeling based on data from 2020 to I think 2000 to 2020. So in that case, predictions from that data, it is possible to compare with the data which we have right now, like maybe from 22 or 23. So have you seen any instances of a correlation between predicted data and future data, and how far has it been able to predict accurately? Like how have you seen the accuracy percentage? How is it?
Yes, I think it's an interesting question, because I don't think scientists like going back and and redoing that and just to prove their point, essentially. So studies are done, published, and people sometimes forget about them. It's possible, and I think you're right. I can think about the oceanographic studies or something like that. They try and predict and hangcast and validate those future casts, I guess, essentially. So I think climate and oceanographic studies are mostly doing that. I'm not aware of any species distribution modeling studies that would do that, basically, because again, you model train on the raw data, you would probably, again, you would have to still sample. I would sample regardless. I would still sample one more time. Once you sampled again, you're wrapping your N on the actual replications. So you will improve your model regardless.
So I think it will be constantly improving it, so the predictions will get better and better, kind of like that.
Theoretically, yes. So if you have more N, you have a better data as well. In the next decade, we probably will have better data sets. Science is moving all the time, developing all the time. So the methods that were applied back 10 years ago probably not valid anymore. Think about machine learning versus linear models. Most of this was previously initially done with something like linear models. These days, very little people would probably do that in that sort of setup. But yes, I think it's fine. It's fine to get different. It comes to that temporal essentially resolution, right? If you predict one time this, the next time you predict that doesn't exist there anymore. Is that true? Is it not? How do you address it? It all depends, like case dependent, I would say.
Thank you.
Thanks. Sure.
If I can add to that, just one spanner perhaps, is that a lot of the statistical approaches to SDMs assumes a stationary relationship between the environmental drivers and the presence of that species. And I might be wrong, I'm certainly wrong, but I haven't seen work that will integrate, for example, adaptation. And the quicker that demography happens, the turnover of individuals in a population, the quicker that local populations can change their relationship to their physical environment. So future predictions to 2050, 2100 are explicitly assuming that the relationship between those environmental parameters and where the species is happy to live does not change. So incorporating any type of adaptation, and the genetic literature is full of that now. So it's just a bit of a precautionary tale that sure we can develop the models, but would you believe them? Maybe.
Yeah, fair point. Sure.
I'm working on species distribution modeling on already. So, in respect of presence points, I have taken the nesting sites along Orissa. So, few of the points are in along the coastal sites and few are in the mid of the land. As you suggested in during session, you have set the geometric extent and you remove the points which are out of context. By the way, any other way to remove the particular points?
Yes. For what's the reason you're removing those for? Just because they're not in the right location, or?
Nesting sites always will be on coastal side, but the point is on mid of the.
Right. Yeah. Oh, absolutely. So you can definitely. It comes down to exploration. So how you identify which point belongs or doesn't belong. So you can just, put like a label each one of them, and remove by a label. So that's a unique identifier. I presume you apply those for your data set. Like it's any type of database management, you will have a unique identifier for each one of your samples. So you can find one of those and remove it by that just here programmatically, or in GIS if you're using any type of GIS software like QGIS or ArcGIS, you can just select the point and just remove that, or export the database without those points. So that's another option. In here, as I said, it depends. I would probably, if not the lazy way, I don't know if you caught that one, which again, if it doesn't have data, then NA remove, that's the easiest one to remove. Otherwise, I would just say, well, clearly that longitude range on land, or latitude for that matter, doesn't matter which one, doesn't belong. So I would be removing by something like that within the data frame sort of space. We'll just say if the latitude is, longitude in this case, above or below a threshold, then that doesn't belong there, and you would remove that way. At least these. Is that okay?
And also, I have implemented in Google Earth Engine. So pulling the data, environmental variables, I have consistency, surface temperature, bathymetry data, and also chlorophyll data. So pulling from different sources, I felt data compatibility issue I have faced. In your perspective, how we can address this?
So filtering for?
I pulled from OBIS database, NOA database I have pulled. I'm asking if one particular data is supporting the other data also shows some issues. Data compatibility issue I faced in Google Earth Engine while implementing this modeling. So any other alternate way to, is there to address this compatibility issue I'm asking?
Sure. So how many layers do you pull? Just the point data or also environmental data? Point data. Point data you can basically, if you use anything like Python, you could, or R for that matter. So again, you can just do it in one of those softwares. Google Earth Engine also have the GE map, I think it's the package that called. So you can, instead of doing this in Java, if it's harder, so then that will be the one. But if the data doesn't work, it's not necessarily the software issue, it's probably the data issue. So there could be something to do with the quality of the actual data sets. If you're not entirely sure about your point, I wouldn't include it in your modeling. So if you see that there is something fishy going on with this, I would probably exclude it, unless you're really limited with data. And that could happen easily as well. No worries.
Can I just... Sorry.
Okay. Sorry, I'll get back to you. Web gives seasonal or monthly biochromatic variables data. I'm not aware of this. I don't know definitely for this area particularly. I do think we may be able to have something like that in Australia for local climate, not across the entire continent though, or the sea for that matter. Yes, short answer, not aware of it. Probably requires some Googling. Yes?
Going back to the turtle issue, are we, should we always limit ourselves to geographical variations like you were talking about SST and the longitudes? Can we use parameters like maybe coastal vegetation that might have come up after some time, so it stops the turtles from, like coming ashore to inshore? So they'll probably restrict themselves only to the sandy areas. And it also depends on which time the data was collected about their occurrences. If it was maybe a year or two before, earlier, then it's possible that there's some vegetation which has developed in that area that's restricting the turtle occurrence.
Yes.
So
I think empirically you need to know what are those areas and how much of that, I guess, abstracts it. The easiest way to incorporate some of this would be through masking. So masking means you exclude some areas of your predictions based on a particular reason. So for example, your vegetation, if you say they don't go on that area, so you just can clip your original raster or final prediction even by that area, so to exclude it essentially. So you predict everywhere else basically. So it comes to the point where also you can specifically generate absence points for those areas. So if you know that that particular, I don't know, dune or beach covered by vegetation that precludes turtle from going onto there, you can just specifically allocate an absence point for those areas, but that's meddling with the data set in the first place. But it's a valid absence point as well. Alternatively, as I said, I would just obtain the vegetation layer. Before or after your model fitting, I would just clip to exclude that, and that will be the easiest way to do it, the lazy way.
Trying to say that there is a possibility that they could have been, present inshore as well. It doesn't have to just because of the it looks like an outlier on the data on the map. It doesn't have to mean that there was no turtle. There's a good possibility it was.
Yeah, absolutely. Yes, totally. Depends obviously. Like from my perspective, that was a fish, so there is absolutely no way it will be inland unless somebody bought it and brought it there. With turtles, yes. With other species, absolutely. So that's the point of know your data. Make sure you use the most logical approach to your data specifically. Wrap it up.
Thank you so much, Dr. Ronan, for laying out all those foundational...
Thanks, guys.
Okay, with this now, I would like to hand over the remaining time to NCCR and AIMS representative to give concluding remarks.
Good afternoon, everyone. So today we end the sessions for the participants, and we would like to hear your feedback from all of you. So what do you think we could improve, or what is that you liked about the session? So anybody who is interested could give your feedback.
I think overall it was really great, ma'am. I had so much to learn from, because from my perspective, I didn't know even some of these, like some of the researchers and their perspectives, what they said, how things could be done. I haven't heard so much of it. So it was really eye-opening for me. So I think my thinking has widened. So because I come from quite a different background. I did my bachelor's in agriculture, and I'm basically from a biology background. So to learn more about pollution and how the marine debris moves and like how it behaves, so that to apply it on biodiversity. So I understood like you have so much to learn about marine debris so that we can understand what are the consequences, like the steps of what are happening, like a cascading of events that we could think of and then make sure to conserve our biodiversity. So it was a lot of information. So I'm still trying to process all that. And I had time to interact with a lot of amazing people. They were kind enough to listen to my doubts, answer them, and I think it was very, very informative, very, very engaging, and in short, it had a lot of things. I thought it would, when I entered, I thought it would be completely away from what I would be expecting. It had a lot more than what I expected. So it was really great, ma'am. 3 days were entirely amazing, and I thank you for, opening up invitations and letting us participate in this. It was really nice. Thank you.
Feedback form.
Feedback form, and we'd like you to scan and give your feedback to all the participants if you don't want to say anything now. Yeah, this is yeah. Yes, yeah. So anybody online would like to give your feedback?
I guess I'll just mention if we have a chance. Am I audible, ma'am?
Yeah, Jaya, you are audible. You can go ahead.
Yeah. The past 3 days, it has really been a life-changing thing for me. Why? Because my area of I am basically from EC department, so I'm working based on satellite-based data, remote sensing. That's what I'm doing, and I don't have much base in that. These 3 days was really very informative, and I gained too much knowledge. And everybody was patient enough even to answer the questions that were asked online. And I'm so happy about it. And I really want to thank everyone who gave me this opportunity. Thank you so much.
If I may just quickly add, for the younger generation in the room, so we are tomorrow trying to also host an event of early career scientists. You could be anything from anything really, and doesn't matter how long you feel you've been in this profession, you might still feel like an early career. So tomorrow we still haven't decided exactly what time, right? But yes, in the afternoon.
Yes, we can.
Yes, but please join us. It's going to be informal. We're just going to have chat and give an opportunity, an additional opportunity for all of you to ask questions from all of us, and more so, and answer our questions as well. Just about, career progressions, career choices, how we end up where we end up. We have a range of scientists here. We invite anybody from NCCR senior colleagues as well to participate if they're interested. We have a colleague here from Indonesia as well, can provide a regional perspective. So please come tomorrow. I hope to see you there.
once again, and before we leave, we can take a photo, group photo, and then we leave. Yeah, thank you so much.
Please come forward for group photo. We'll have a group photo. Close the session. Okay.